Start the first session. It's my pleasure to introduce Dr. Yvonne Perry from the University of Strathclyde, and she's going to give us her thoughts on non-viral delivery of mRNA.
Okay, thanks. Andy, that was a kind introduction, really useful setting the scene for us. So as Andy pointed out, there's a range of delivery systems we want to look at for RNA. And we've been looking a lot at that in the work we've been recently doing, funded through a European Union grant. So.
We see you fine, you're good to go.
Excellent, thanks Jane. Okay, so just bringing us back to what Andy was saying, nucleic acid vaccines are been around for a while now, roughly 3 decades, where we started with the DNA vaccines, and then we've progressed on from DNA to the RNA and also the self-amplifying RNA, with the various advantages. When we move from DNA to RNA, we don't need to get into the nucleus, and then the self-amplifying improve potency. But with all of these systems, all the nucleic acid systems, we still need to have a delivery system because the RNA is highly anionic, it's highly hydrophilic, so it's very difficult to get into the cells. It's also easily and quickly degraded by enzymes, so RNAs. So we did quite a lot of studies, and we look at a range of delivery systems. Now there's more than I'm going to outline here available, but I just thought I'd show you the ones we've been really looking at. And that's the conventional, I would say, liposomes, where we still have an aqueous core, lipid bilayer, and you can put the drug in the aqueous core or absorb it onto the surface. Then we have the lipid nanoparticles, which the definition between the two is still quite grey, but basically when you put the RNA within the structure, it does restructure and you lose that aqueous core and it condenses down. So obviously these have a cationic element to it. We can also have solid lipid nanoparticles, and we've looked at those, and those have a core of solid lipids, so it's a high melting point lipid. So kind of like a polymer nanoparticle, but the inside is lipid based. And then we have our polymeric nanoparticles. Generally been looking at those based on PLGA. And finally, we have a emulsion type system. So we've been looking at cationic nanoemulsions as well as the final option. So those are the 5 platforms we've been investigating to compare and contrast how they look. And basically, just to remind you that LNPs are not new now. We do have a product approval. There's more than one now, but we know that Impactor was licensed in 2018 by the FDA. And that's an ionizable lipid nanoparticle system. So for all our protocols, we've been using the same platform. So we've been using the nanoassembler. And this is an outline schematic, basically, of how we've been making the LNPs. There's some variation in how we make the various different nanoparticles, but it's pretty much the same format, where we have an RNA in a citrate buffer, we have a lipid or polymer in a solvent. We then put it through the nanoassembler. We've been removing the excess solvent using either dialysis or PFF. That would also remove non-incorporated RNA if we had any, but we do get high loading. And then we have a purified product. So in terms of platforms we considered, so we have liposomes, and we made them either with the cationic lipid DOTAP or the cationic lipid DDA. So you can see the structures at the side of the screen. So these are both easily available, commercially available, and that's why we chose those. So we made our liposomes with DOPE and the cationic lipid, a 1-to-1 weight, and we either put the self-amplifying RNA on the surface, or we made it in situ so that the RNA was inside. We then did similar. We made solid lipid nanoparticles with tristeran. Again, we either use DOTAP, DDA as a cationic lipid component. Similarly, we made polymeric nanoparticles using PLGA 50-50 ratio. Again, putting the RNA on the surface or inside because we added either cationic lipid. And finally, we tested the emulsion system. So obviously with the emulsion, you can't put the RNA inside, so it was only surface absorbed. And what we did, we had a screening process where we looked at the physical chemical attributes, cell uptake, and then in vivo response, and actually biodistribution as well. So it was a 4-stage process. And we just published this. This is work that just was published in May 2020 in Vaccines, so the full paper's down there. So when we looked at the physical chemical attributes, I mean, obviously we can control these and how we manufacture it. So, but we were really aiming for below 200 nanometers, ideally around about 100. So you can see we were able to produce the liposomes, and I've generalized the data because it was a lot of data, but Really didn't matter that much whether we had DOTAP or DDA. The systems were always cationic. Because they were always cationic, we always got great loading because it's an electrostatic interaction. They were always able to protect the RNA from enzyme degradation, as we would hope. And then the sizes fluctuated slightly, generally with the liposomes slightly smaller in size, and we could produce them even smaller if we wanted, but we're trying to get them all roughly in the same size range, so it was more of a head-to-head comparison. So you can see they're all about 100 to 200 nanometers in size. So the first thing to do we looked at was in vitro screening. So we were looking at toxicity, obviously, to limit we can have on that. We then looked at the uptake, which with cationics, is it uptake, is it surface absorption? It's difficult to tell, but we looked at association, cell association we call it. And then finally we looked at expression of the self-amplifying RNA, and we used luciferase tag for that. So we were looking for expression of luciferase. And we used BHK cells. So here's the data. So I appreciate there's a lot of data here, but basically I'll summarize it down for you. So you've got your DOTAP formulations and your DDA formulations. DOTAP tends to give us a higher cell viability, so we can work at higher concentrations compared to DDA. But we always made sure we were working in the cell viability range. So our next set of graphs, where we see mean fluorescence, this is cell uptake / absorption association. And basically you have all the different platforms below. So you have our liposomes with entrapped SAM, liposomes with SAM in the surface, SLNs, solid lipid nanoparticles inside, outside, and then polymeric. So there was a bit of a problem with our PLGA systems. We could only make them with SAM and trap when we used DOTAP, and then with DDA we could only make it in the surface. We got some aggregation. But in general you can see that in terms of cell uptake, with the DOTAP or cell association, there's really not that much difference that you would be excited about. With the DDA, you do see the polymeric nanoparticle giving higher cell association uptake, so that stands out. But then if you look at the cells that have been transfected, so these are actually rabies transfected cells we're looking for, the antigen for the rabies virus. Really, if you look across the platform, there's not that big a difference. Nothing that would excite you and tell you, woof, that's the one to go for. So you can really see that from our in vitro studies, we weren't getting a good handle on what is the lead formulation we should be going for. So there's no notable standout protocol. So that tells us it's quite difficult with our protocols to figure out what should go in vivo. But we did have to do a down screening process. So we decided to go with 2 liposome formulations. So we have a DOP DOTAP and DOP DDA. So we could head-to-head comparison. And then we also took our PLGA nanoparticles, which have DOTAP in them. So we then vaccinated mice twice on day 0 and 28, groups of 10 mice. They were vaccinated with either 1.5 or 0.5 micrograms of self-amplifying RNA encoding for the rabies virus glycoprotein G. So we chose that because there is a commercial product vaccine available that we could benchmark against. And then we looked at antibody responses and also cell responses. So what we have here is our rabies anti-IgG titers. So, and if you look at the graph, the dotted line at the bottom of the graph is basically our limit of detection. So anything below that, which is very low, is basically we aren't picking anything up. The red line, slightly higher up, where it says greater than 0.5 EU per ml, that's an indication of protection. So if we see response above that, then we have protection based on this. And what we have tested is our DOTAP nanoparticles, NP, at 2 different doses, 1.5 and 0.5. And then we have our liposomes or LNPs, depending how you define it, because the RNA was inside. Again, 1.5 and 0.5. We also have an emulsion system, the DOTAP emulsion system. And then Rabipur is the commercial vaccine, which is not RNA based at all, but that was our benchmark from it. So you can see this is a response, 43 days. They've had 2 vaccines, 2 doses. It's 10 mice averaged out. And what you can see is really that our nanoparticles and our LNPs are given similar responses. There's no significant difference, all above the threshold. We don't really see a dose response, so the 1.5 and 0.5 are very similar. and we don't have a significant difference there. We do see our emulsion systems giving us higher response, and our Rabipur, which is a benchmark, is giving us higher. But in terms of the delivery platforms, we really couldn't pick out a notable difference between our nanoparticles and our LNPs, and also we couldn't really pick out a notable difference between the DOTAP and the DDA. So that was interesting. So it's telling us that the delivery platform is not jumping out as much as we might have thought. We then looked at cell-mediated responses. So I know there's a lot of colour going on here, but basically you're left this graph, the CD4, you're looking for as high a response as possible, and CD8 again. And what you can see is our nanoparticles really stick out more than our liposome formulations in terms of CD4. And there's not any significant difference in terms of the nanoparticles and the emulsions in the Rabipur. So really, that would suggest these nanoparticles are jumping out a bit more as a potential delivery system, which was very interesting because generally everybody has known that LNPs are a strong profile. And in this case, we weren't seeing that. So we thought, oh, that's interesting. We need to go back and rethink this again. So we then looked at our LNPs a bit more, and we then screened a wide variety of cationic lipids. So we actually took 6 different ones, and these are still all cationic in nature, so they'll always have a fixed positive charge. We also did benchmark against the ionizable lipid, the same one, the DLN formulation, and we had basically 6 lipids. We then formulated them either without DSPC, so they're just basically DOP, cationic lipid, and the PEG, or we put in the DSPC as well, so to mimic the formulations that Andy was actually talking about early on with his studies. So again, this was what we have, it's actually just out, but it's still in the proofs concept. I don't think we've got the corrected proofs done yet, but it's online now. So again, screened a lot of formulations, and I'm not going to dwell on it too much, so you can see all the different cationic lipids we benchmarked, and here's our ionizable lipid we compared against. And basically we looked at them all, and generally they're all below 100 nanometers, all cationic, all giving us high absorption. Sorry, all nearly neutral, even though they have a cationic because we've got the PEG in it, and giving us still high loading. So how to select was quite difficult. We down selected the DS tab because they were quite difficult to work with. So we decided to go with DOTAP DDA to give us a comparison, and we benchmarked against the ionizable lipid formulation. Exact same vaccine protocol again using the rabies G protein. So here we have the IgG responses. So this is 2 weeks after the first injection. So they've only had 1 dose here. So what we now see, again, the bottom line is the limit of detection. The upper line is the limit of efficacy. So you want to be above this line. And now we have our benchmark Rabipur again, which is a 1 in 20 of the human dose. We then have our DOTAP LNPs, our DDA LNPs, our emulsion, and our benchmark ionizable lipid. And what we can see here is, again, we're not really seeing a dose response between the 1.5 and 0.5. Equally, we're not seeing any significant difference after one dose between the DOTAP, the DDA, and the emulsion. And at the higher dose of 1.5, the cheaper cationic lipids give the same efficacy as the more expensive ionizable lipid. If we then boost it with a second dose, all our responses go up. They go up about 20-fold, and you can see that shown here. But you see a very similar trend, that we don't see any notable difference between our cationic DOTAPs, our DOTAPs LNP, our DDA LNP, and our ionizable. Only at the lower dose of 0.15 does the ionizable formulation give us higher responses, but in all cases, they do give us protective response. So that tells us we can use the cationic lipids, but our ionizables have a slightly enhanced potency compared to the non-ionizable. So we also did look at biodistribution. So, sorry, let me go back. So we also looked at biodistribution. So we compared the cationic LNPs made with DOTAP, DDA, and the ionizable lipid. And what we've done is fluorescently tagged the liposomes. So we're looking at where the liposomes or the LNPs go, not where we're getting expression. And you can see over time, if you go down the columns, you can see it's saying at the site of injection. And if you look at the graph for total flux, what you can see is this is us measuring the clearance of the LNPs from the injection site intramuscular. So what you see is the DOTAP and DDA have different clearance patterns, but really for a vaccine, they were given the same response. And also you can see that the DOTAP cationic LNPs versus the ionizable LNPs have different profiles, but actually gave us very similar response. So whilst you are getting a different pattern of biodistribution, basically it wasn't giving any usable insight into what was a better vaccine in that respect. So biodistribution profiles are interesting, but they aren't telling us anything we can use as a predictor of a good vaccine. But we do see them all forming a depot, whether they're ionizable or not. So that's useful to know. It is staying at the injection site, but whether you still want it to move is not clear yet. So in summary, we have tested a range of platforms, and the liposomes give a quite a good response. Our LNPs give a strong response, but also our nanoparticles in that respect. Our polymerics were giving us a good response. So there is optionality in delivery system to look at, but in all cases that we were able to make them easily using microfluidics, and we can scale up in reproducible format. So that was really interesting. And just to acknowledge this, as usual, as you would expect, was a big team involved. A lot of this work was funded by the Fast Train Vac Consortium, which is an EU Horizon 2020, and a lot of the in vivo data, all of the in vivo data actually, was from Julia and Gustavo, who did the work, along with Sini, who did the IVIS, and the rest of our core team are here with Cameron, Gillian, Neil, Rachel, and Carla, all helping to support this work in various ways. Thank you for your time.
Thank you, Yvonne. And your timing's good, 15 minutes, so we've got 5 minutes for a few questions. Jane, is somebody gonna ask the questions?
Yes, Shell's going to join you, and
Perfect.
he can ask a couple questions.
Sure. Hi. One of the questions was about generally how you formed the nanoparticles. There were several questions about that, so maybe if you can touch upon your basic techniques, Yvonne.
Mm-hmm. So in all cases, we use microfluidics for that. So we would always be dissolving our polymer in a solvent. It depends which one. So some were easier dissolved in some solvents than others. So we might have to use like ethanol for our liposomes. We were using like acetonitrile for some of the PLGA systems. So it did vary. So we had to optimize the solvent for the given component we were trying to dissolve. So, and it was similar with LNPs. And the emulsion system we did use with the nanoassembler, but equally we also just went with a standard emulsifier for that system. So that was the kind of key thing. Removing the solvents, we use dialysis mainly, but just for quickness, but we have optimized for PFF.
Okay, thank you. There was another question about whether or not you performed any in vitro data with cell uptake in full human plasma.
So yeah, we did it with 5% fetal calf serum and serum free. So we looked at both and we didn't see much difference. And I think it's probably because at the moment we're not discriminating between cell uptake and cell absorption. So because they're cationic, they go down and they just stick onto anything. So we get high association. We really need to go back and do a study where we do it at 4 degrees and see if it is absorption or uptake, because that would discriminate more for us. But actually, given that we didn't see any usable differences, looking at with and without FCS, and we didn't keep going with it. But that would be something we don't know yet. Is it just stuck in the surface or is it uptake? So that would be something probably we need to explore more.
Thank you, Yvonne. Another question about the controls for your experiments. Some people are asking how how encapsulated systems might compare not only with each other, but also with naked RNA. If you can comment on that.
Yeah, so we didn't try naked RNA because generally a lot of the previous work has shown that you don't get a response. So I think that was quite established early on. So we didn't include that control just for refinement, reduction and replacement, you know, number of animals you use. So we've never tested that. What we were looking at was basically a head-to-head between the different formulations. And I think, Andy, your original paper, you showed that without any delivery system, it doesn't work. So
it does kind of work, but you get a huge variability in vaccination, and you need to use quite a high dose. And when you boost, some of the mice boost, some don't, but there's a lot of non-responders. The moment we put it in a non-viral delivery system, all the mice responded, all the mice boosted. And so we quickly moved away from naked delivery. I think the reason why we show that RNAs is upregulated during vaccination extracellularly, and the naked RNA just gets degraded quite quickly. The delivery system's capable of protecting it. I think we have 1 minute left, so I'm going to ask the last question to you, Yvonne. So if I'm an academic lab, I'm focused on antigen design, and I've got mRNA being made. It looks like I don't have to go out and get a license to a complex proprietary delivery system. If I wanted to test my antigens in a mouse model, I could read your papers, your work, maybe get a nanoassembler or an Ignite system in the lab and make formulations and do vaccinations and kind of get an understanding for my core technology, which is antigen design. Yep, I can benchmark and see how my antigen designs change the immune response in the mouse. Would that be a fair comment?
Mm-hmm. Yeah, definitely. And that was really where we were coming from, because we know there's a lot of proprietary lipids that will be very good. But if you just want to test your system and get going, you want off-the-shelf products, you want to be able to publish, you know, as an academic and get some sort of feel. Yeah, you can go and just buy DOTAP from any of the suppliers, go with the standard formulation, and then we get a good response, DOTAP or DDA, and we didn't really see a discriminatory between them. And I think DOTAP's a bit cheaper and easier to get, so supplied from Sigma. So that was the kind of premise of our work. We wanted to make it accessible, as you know, that's kind of one of the points of my mantra of my research, is trying to make it globally accessible research.
Well, thank you for all your insights, Yvonne. I think you're going to be very busy with a lot of collaborations coming in from around the world, and I wish you all the best. And with that, we'll say thank you to Yvonne, and we'll move on to our next presenter, Joy Ita Roy, who comes from NEB, and she's going to walk you through the anatomy of an mRNA as a prelude to the session that NEB and TriLink are going to give you on, you know, everything you need to know about making mRNA. Over to you.
All right, thanks, Andy. Jane, can you hear me okay?
Yep, you're perfect. Everything looks great.
All right, thanks, Andy, for the introduction. Hello, everyone. I'm Joy Roy from New England Biolabs. So what I'm going to do for this session is introduce a little bit about mRNA, talk about what is a messenger RNA, what are the key structural features of an mRNA, what does the life cycle of a natural messenger RNA look like, and based on what we understand about the biology of natural messenger RNAs, how can we actually design a synthetic mRNA molecule that will result in optimum functionality in vivo? So let's take a step back and take a look at the flow of information in the cell, the central dogma of molecular biology, where the genetic information is stored in the DNA, which needs to be expressed as proteins, which are the final effector molecules in the cell. Now, this flow of information from the DNA to give rise to the protein involves an intermediary molecule, the messenger RNA. And this mRNA contains the information from the DNA, which is then used to encode the protein by the cellular translation machinery. So mRNA is an intermediate molecule, and its expression is transient in nature. Therefore, if it can be delivered into a cell, you can easily imagine that the expression from the mRNA will be quite rapid because it does not have to go into the nucleus, get transcribed, and you can express the protein of interest directly from this messenger RNA. Now, this is easy to titer, and from a synthesis and manufacturing perspective, if you have the sequence of the DNA, you can essentially synthesize any messenger RNA and express any protein of interest using the cellular machinery. And, you know, this generality of this approach, it's a simple process. It makes it quite lucrative. Now, even though this cartoon of an mRNA that I'm showing you here is quite simple, the actual life cycle of an endogenous natural messenger RNA is quite complex. And it can actually be broken down into multiple steps. And each one of these steps is highly regulated, and it actually affects the final outcome from the mRNA molecule. So in a nutshell, the RNA is transcribed in the nucleus. It undergoes modifications at its 5' and 3' end. It also undergoes editing and splicing, and then this mature mRNA molecule is transported or exported into the cytosol, where either it undergoes translation to give rise to the protein of interest. It can also undergo degradation, where the half-life of this messenger RNA can get reduced. And under certain circumstances, this messenger RNA is also subjected to transport and localized in specialized structures. Now, this balance between translation, where the protein is expressed, decay of the mRNA, as well as its localization is actually really important, because the extent of expression from that mRNA is going to be dictated by the coordination of all of these events. And as you can see, all of these individual steps, right from the transcription all the way to its translation and decay, is actually guided by a number of different interactions that take place between the messenger RNA and a lot of different cellular factors. Now, interaction of the mRNA with the cellular protein factors actually determines the fate of the mRNA. And there is a constant competition between binding of either the protein synthesis machinery, the translation factors, or the decay factors to this messenger RNA. Now, the presence of a 5' cap binding protein at the 5' end of the mRNA, together with the binding of a poly-A binding protein at the other end of the mRNA, it actually results in the formation of a translation competent ribonucleoprotein particle. And this actually results in efficient expression from this messenger RNA, and also prevents its degradation, because it does not allow the decay factors in the cell to bind to the messenger RNA. So what is really dictating these different interactions between the messenger RNA and all these different cellular factors? And this answer actually lies in the structural features of the mRNA, which are the main determinants of these different interactions. So most eukaryotic cellular messenger RNAs are modified at the 5' end by addition of a 7-methylguanosine residue. This 7-methylguanosine structure engages with critical translation factors, and it actually helps in recruitment of the ribosome to promote translation. On the other hand, the 3' end of the mRNA is characterized by the presence of a poly-A track, and this poly-A track is recognized by poly-A binding protein that helps in the formation of a closed loop structure via interaction of the two ends of the messenger RNA. Now, as for the body of the mRNA, it is also divided into distinct parts. So the main body of the mRNA consists of the open reading frame, or the coding sequence. And this is the actual sequence that defines the protein that will be synthesized from the messenger RNA. Now, in addition to the coding sequence, you also have sequences upstream and downstream of the coding sequences, and these are called 5', which is the upstream sequence, and 3' untranslated regions. So these are not coded into protein, but have regulatory functions in the cell. And these regulatory sequences that are present in the UTRs can either accelerate translation or degradation and regulate the fate of the messenger RNA by mainly altering its interaction with the different cellular factors. Now, some of the mechanisms by which either the stability of the mRNA, that is the half-life of the mRNA, or the translation, that is the expression from the messenger RNA, are altered by these different structural elements have been highlighted here. And you can imagine that it consists of a complex set of interactions, and these are dynamic in nature, as a result of which they impart this another complex layer of regulation of expression from the mRNA. This is further complicated by another layer of regulation that is imparted by chemical modifications of the mRNA, the base modifications that Andy alluded to early on. So chemical modifications in the RNA can be present at the end of the RNA or the body of the mRNA. One of the most common modifications of the messenger RNA is present at the 5' end, the 5' cap of the mRNA. It's really well studied and it's well documented structure that is post-transcriptionally added to the mRNA in the nucleus. And the mRNA in higher eukaryotes actually have a very, very similar structure. So it consists of a guanosine, which is then methylated to form a 7-methylguanosine structure. And this is done via 3 different enzymatic steps. Now, this 7-methylguanosine is linked to the first transcribed nucleotide in the mRNA via a 5' triphosphate bond. And this results in the formation of a minimal cap0 structure in lower eukaryotes. However, higher eukaryotic messenger RNA can also have further modifications. There can be an additional methyl group on the ribose 2' position of the first transcribed nucleotide, giving rise to the cap1 structure. And the adjoining nucleotide can also be subsequently methylated to give rise to a cap2 structure. Now, these modifications of the 5' cap of the mRNA is really important because it acts as a platform for multiple mRNA-protein interactions. So the cap1 and the cap2 structures are important because they distinguish between self and foreign RNA via the altered recognition by cellular sensors that are known to induce interferon signaling. Furthermore, some vertebrate messenger RNAs, about 30% of vertebrate messenger RNAs have been shown to have other modifications at the 5' end. They can either have M6A or 2'-dimethyladenosine, M6AM, as the first nucleotide. The M6AM modification is formed by methylation of an adenosine nucleotide that is right next to the 7-methylguanosine cap. And some studies have actually shown that the presence of this M6AM signature in the messenger RNA actually increases the stability of that mRNA, and it's also implemented by altering its interaction with some of the decay factors in the cell. Now, most of the modifications, the chemical modifications in the cells were first discovered in non-coding RNA. However, because of the recent advances in detection methods, it has now been demonstrated that the body of the mRNA can also have chemical modifications. And these are present not just in the untranslated region, but it can also be present in the coding sequence of the messenger RNA. Now, what are the exact biological functions of these base modifications? It's really not understood, and it's really complicated to tease it apart. However, you can easily imagine, because these modifications can alter the mRNA structure, it can alter its interaction with cellular factors. So the function of these could be quite complex. So currently, a number of different chemical modifications have been identified in endogenous messenger RNAs, and some of those have been highlighted here. Now these are important because the idea here is, if you're using a synthetic mRNA as a drug, what you would really want to do is synthesize an mRNA molecule in a tube, and you would want that synthetic messenger RNA to recapitulate what exactly happens in the life cycle of a natural mRNA. You can bypass some of the steps because the transcription is, let's say, done in vitro. But the idea here is you would want that synthetic messenger RNA to interact with the cellular components so that it can express the protein of interest by hijacking the cellular translation components. So today I'm not going to go into the, in this session, I'm not going to talk about the actual synthesis process, because we will have a breakout session where we will describe in details how messenger RNAs can be synthesized in vitro. And I welcome you to join us in that session if you're interested in making messenger RNAs in a tube in the lab and try to see how these messenger RNAs can be used for different applications. Now, the main idea of the synthetic messenger RNA is that once it is delivered in vivo into the cell, it should be able to interact with the same cellular machinery to express the protein of interest. However, if we look at the life cycle of a natural endogenous messenger RNA and compare it to what happens when a synthetic messenger RNA is introduced in a cell, you will see that there are a lot of differences. So the endogenous messenger RNA is transcribed in the nucleus, and then it interacts with a lot of different cellular factors and exported into the cytosol, where it then comes in contact with either the translation machinery or the degradation factors, and that results in the expression from the messenger RNA. Now, on the other hand, a synthetic mRNA molecule here, based on the delivery route that you're choosing, could actually go to the endosome. And what it does is it then starts interacting with some of the other cellular factors that a natural messenger RNA never encounters. So it could interact with immune receptors that are otherwise not encountered by natural endogenous messenger RNA. And this is important because for certain applications of synthetic RNA, you do not want to trigger an immune response. And what you want is that the synthetic mRNA molecule that you're introducing should be recognized as a self molecule in the cell. It should reach the cytosol, interact with the translation machinery, and result in the protein expression. Now, there are a lot of different immune receptors. There are multiple immune receptors that have been demonstrated to be activated by synthetic messenger RNAs based on the localization of the mRNA molecule, and some of them are highlighted here in the slide, and their corresponding ligands are also highlighted. So it is really critical that we address the requirement of increased expression and stability from a synthetic messenger RNA, as well as its reduced immunogenicity, again, for certain applications, not all applications. And in the next few slides, what I will do is highlight some of the approaches that are generally being implemented to increase the stability and expression from synthetic messenger RNAs, and therefore express the protein of interest and reduce its immunogenicity by altering the sequence and the structural features of the mRNA. So this actually can be done in a couple of different ways, and some of them are highlighted here. And there are three different criteria that you would want to keep in mind, or you would want to consider while designing an mRNA. And some of these are highlighted here. So these are important to ensure that you are getting optimum response from your synthetic mRNA molecule in vivo. Some are important for expression from the mRNA molecule. Let's say, for example, the length of the poly-A tail, the actual sequence of the 3' untranslated region, the length of the UTR, and also whether or not you have certain stabilizer elements in your UTR sequences. So if your application, if your synthetic mRNA application requires the stable expression from a messenger RNA, if you need prolonged expression, these are some of the criteria you might want to consider and might want to introduce or install in your synthetic mRNA so that you can recapitulate how the cellular machinery actually uses these in natural messenger RNAs. Some of the other things that are important is, let's say, for example, the uridine content, which affects the immune response from the synthetic mRNA molecule. So it's mainly because it has been demonstrated that some of these immune receptors, the toll-like receptors, recognize U-rich patches in mRNA molecules. So if you do not want to induce an immune response, you might want to deplete your messenger RNA from these uridine-rich sequences so that you can overcome or overwrite the recognition by some of these sequence elements. Now, the other aspect that you might want to consider is changing the composition of the messenger RNA so that it looks more like a natural mRNA. And this is based on tons of work, and I'm just going to highlight a few aspects very briefly. So introducing modifications in the mRNA cap to reflect endogenous messenger RNA structures, let's say introducing M6AM modifications are being evaluated, and this can definitely change the expression and stability of your synthetic mRNA. As I mentioned earlier, poly-U patches in the mRNA are recognized by toll-like receptors. Therefore, it was postulated that replacing the uridine nucleotides in your synthetic mRNA with naturally occurring uridine analogs, let's say, for example, pseudouridine, could actually prevent recognition of the synthetic mRNA by these immune receptors. Furthermore, introduction of mammalian mRNA was found to be less immunogenic as compared to bacterial or mitochondrial messenger mitochondrial RNA, and it was mainly thought because mammalian messenger RNAs contained modifications. And because of this, chemically modified messenger RNAs are heavily evaluated in depth, and there, as Andy alluded to early on, there's a specific class of messenger RNA, these chemically modified messenger RNAs that are being investigated for a lot of different applications. So why are modifications important? And I'm just going to summarize the data that has been seen by a lot of different groups. Similar to everyone else, what we saw was if you introduce these modifications in synthetic mRNA, let's say if you replace uridine with pseudouridine, you can actually increase the expression from your synthetic messenger RNA. And at the same time, you can also decrease the immune response that you see from these messenger RNAs, and therefore these are routinely incorporated in mRNA for optimizing expression and reducing the immune response. Now, the modification in synthetic messenger RNA can be incorporated in a couple of different ways. You can do it by replacing or completely substituting one standard NTP, in this case, let's say uridine with pseudouridine. Or what you can also do is do a partial substitution where the reaction has a mix of modified and standard NTPs. And based on the approach that you take, you will end up with two different outcomes. Now, in case of partial substitution, what you will see is the resulting mRNA molecules will be heterogeneous in population. This is mainly because the modifications will be incorporated randomly throughout the body of the mRNA. However, on the other hand, if you're doing a complete substitution, the mRNA molecules that you will get at the end of your synthesis process will be identical. Now, this might be more, this might be preferred from a regulatory perspective, because if you're using a synthetic mRNA as a drug, you would want all of these molecules to be identical. And because of this, complete substitution of messenger RNA with modifications is currently implemented for most of the programs. However, if you are substituting your mRNA completely with a modified nucleotide, you need to keep one thing in mind, and that is that the natural messenger RNAs, even though they have pseudouridine, let's say, for example, it has been identified in natural messenger RNAs. However, the frequency with which they have been identified in endogenous messenger RNA is very different as compared to what we are introducing in synthetic messenger RNA. So the question here is, what really happens when you heavily modify the synthetic messenger RNAs in the cells? What are the biological consequences of that? And how does the cellular machinery recognize it? And how does it affect the final outcome from these synthetic messenger RNAs? So this has been studied recently by us as well as others. And I am just going to highlight some of the recent findings. Now, based on the nature of the modification, few different outcomes have been reported. Now, incorporation of N1-methyl pseudouridine in the messenger RNA, and I'm talking about complete substitution, it has been shown that it actually alters the secondary structure of the mRNA quite a bit. And because of that, it also changes the ribosome transit time. And the effect of the modification on the secondary structure is different based on where the modification is and what the effect it has on the final expression. For example, if these secondary structure, how the secondary structure affects the 5' UTR versus the coding sequence, and what the effects of those structural changes are on the final expression is quite different. Now, because the ribosome transit time is affected, now some of these modifications can also alter the base pairing. And therefore, it's not surprising that under certain sequence contexts, when you introduce these modifications, these can lead to low frequency translation miscoding events. And we have observed that when you use fully substituted pseudouridine messenger RNAs, a synthetic reporter messenger RNA, you can actually see some really low frequency miscoding events. So therefore, it's really important to keep in mind the final outcome that you're looking for from your synthetic messenger RNA. And therefore, while choosing the approach of introducing chemical modifications in the messenger RNA, those final outcomes need to be taken into consideration. There are a lot of different parameters that will be affected by the presence of chemical modifications. And one thing that is really easy to understand is that based on the sequence context where that modification is, it can have very different effect. So, as I mentioned earlier, that there are sequence features and there are sequence requirements that can be changed to optimize the expression and stability of mRNA. The other thing that has been highlighted is that the actual synthesis process that is implemented to make the synthetic mRNA can also contribute to the final outcome, the final expression from the synthetic mRNA. Now, it's known that the in vitro process by which messenger RNAs are synthesized are not very robust. They do form certain byproducts, and it has been seen that some of those byproducts are deleterious because the RNA structure that you get is very different from a natural messenger RNA, as a result of which these are known to induce some immune response and have detrimental effects. So therefore, purification strategies have been implemented so that you can separate out these impurities from your RNA of choice, and alternative workflows are also being implemented so that you can prevent the formation of these different byproducts. I'm not going to go into details, but just highlight one thing is that in vitro transcribed RNAs were shown to be to activate TLR3, which gets activated by double-stranded RNA, and it's a known ligand of TLR3. So for a long time, it was hypothesized that these double-stranded RNAs in the IVT RNA mix are byproduct of the in vitro transcription process. And this was further confirmed by a study by Catalin Carrico's group. It's a really elegant work where they actually demonstrated that IVT RNA definitely contains double-stranded RNA byproducts, and you can actually implement, let's say, HPLC or a purification method to separate out those double-stranded RNA byproducts from the main RNA, IVT RNA, and that results in loss of an immune response. So this reinforces the idea that IVT RNA, like in vitro transcription of RNA, it definitely forms the RNA of choice, but it also results in the formation of certain byproducts that are not desirable. So currently HPLC is implemented as the golden standard for removing some of these undesirable byproducts. It's not very cost effective, and it's definitely very difficult to scale up. So it's also really important for us to understand why are these byproducts made, and what is the nature, what is the structure, what is the sequence, or what is the source of these byproducts, so that you can design your RNA in a way so that you can prevent their formation in the reaction in the first place. So work by us and others have shown that some of these byproducts can actually be tackled by changing the sequence of the RNA. So it reduces it to some extent. It does not completely get rid of it. So these are some of the things that can be implemented while designing an mRNA so that you can achieve optimum functionality. So these are some of the examples I hope highlights the importance of understanding the biology of a natural mRNA, so that you can alter the structure and the sequence of your synthetic molecule, as well as you can alter the synthesis process so that you can achieve functionality from your synthetic mRNA. And based on your application, some of these will be a little more critical than the others. And with this, I would like to end here. And before I end, I would like to thank New England Biolabs for supporting our basic research on mRNA modifications. Thank you.
Thank you very much, Vaijuita. You gave us a comprehensive overview of the anatomy of an mRNA and your thoughts on structure and function and how we might go about designing our mRNAs. Unfortunately, we've run out of time for questions, but it was a great walk through the science, and people can certainly follow up with questions. You'll be on the panel at the end of the symposium. So thank you for joining us and walking us through that today. With that, we'll move on to our final speaker. This is Lloyd Jeffs, who works at P&I on the GMP manufacturing side. And he's going to talk us through what a target product profile might look like for an mRNA vaccine, and strengths, weaknesses, and where future research is probably going to focus on, particularly on how do we put a low dose mRNA in a vial and get the stability requirements for a commercial product. So with that, I'll hand you over to Lloyd, and he can walk us through. Thank you.
Thank you, Andy, for the introduction. I'm just going to try and share my screen with everyone. I'll make that full screen. So, so for developing an RNA medicine, you really need a framework. And here we've summarized at a high level the basic steps. And that starts with discovery, your molecular target validation, and also deciding on your API or your RNA of choice, and your excipient ID. And for delivery systems, that's typically novel ionizable lipids in lipid nanoparticles. So then the early preclinical work takes place, and that involves formulation development and formulation optimization. Once you've optimized your formulation, you can then move to the late preclinical stage, where you are basically zeroing in and selecting your nanomedicine candidate. And that involves process development work, a number of animal studies, and sometimes in a second species, to assess the efficacy and the toxicity and the safety profile of your drug. Analytical methods are an important part of the late preclinical. You have to be sure that the data you are generating is valid. So there's a lot of work that goes into analytical method development and qualification. Once that's done, everything has to be within a quality management system. And quality doesn't get enough profile at these types of symposiums, so I'm going to give out a shout to quality. Everything you do is basically governed by quality systems. And that's a little bit different from, say, the discovery work that's started at the beginning of the process of developing an RNA medicine, but it's awfully important. And that basically sets you up for your basically regulatory submissions, whether it's a clinical trial application or an IND application, to start the clinical development. And importantly for that, you have your analytical methods qualified, you have your raw materials sourced, you have a GMP manufacturing process, and that's typically done in a manufacturing facility that is a clean room, an established contract manufacturer, or some larger companies have their own capabilities and facilities as well. And if everything goes well through your clinical development, you have basically start thinking about commercial supply, which is quite exciting. And typically this process takes, let's see, between 8 and 12 years, so roughly around 10 years to do this to get a commercial launch. So it's a long time frame. So what guides the development of the nanomedicine? Well, you've got to think about where you want to be in the end. What's your end product? So for that, you have a target product profile. And that defines the required and desirable properties of your product, provides an end goal for the development, and differentiates your RNA medicine from other existing alternatives or standard of care. And it's also important to basically continue updating and briefing regulatory agencies and any partners that are involved in your work. So here I've summarized, or this is the source is the WHO target product profiles for a COVID-19 vaccine. And I've taken excerpts from that guidance that was from April this year. And I think that was upon consultation of many different academic and industrial and thought leaders in vaccines contributed to this. So these are some of the vaccine characteristics, or the important vaccine characteristics, and the preferred properties, or the critical or minimal, in the right column. And you can see, obviously, duration of protection is important. The route of administration is also important. And for RNA medicines or RNA vaccines, the last three are extremely important, and I'm going to focus on those. So for product storage and stability, it's preferred to have higher storage temperatures and higher thermostability, and that will help enhance vaccine distribution, getting it to the sites where it is used. Currently, the challenge is that most mRNA vaccines or products in development are actually stored frozen at less than -60 Celsius. So some work needs to be done on that. Secondly, for presentation, you're thinking about making a large number of vials very quickly. So a multi-dose presentation is really preferred in this case. And you can see here the maximum injected dose is quite small. It's 0.5 of a milliliter is preferred. Can go as high as 1 milliliter for a vaccine application. And accessibility is super important as well. So we want to basically have the capability to rapidly scale up production at a reasonable cost per dose that allows broad use, including in lower and middle income countries. So those are really important considerations in developing your target product profile, in this case for an mRNA vaccine. And just to summarize there, you can see that these are the three main points I want you to take with you. RNA vaccine stability, very important. One of the ways I think people are looking at this already is trying to provide a lyophilized form for RNA vaccines, provide sufficient vaccine doses, and with the new highly potent RNA payloads, the industry is dramatically increasing the vaccine doses per RNA LNP batch. Also considering multi-dose presentation systems, so a vial that you can use multiple times while maintaining the integrity of the contents. And lastly, and probably most importantly, is accessibility. And one of the approaches to this, I think a very exciting approach, is decentralized vaccine manufacturing and having modular, rapid, deployable, in-country RNA vaccine manufacturing. And that's really going to help, especially responding to pandemics such as COVID-19, in taking vaccines that are approved for use as an emergency use or a fully approved vaccine and getting it to people all over the world wherever it's needed.
Okay, thank you, Lloyd, for walking us through, a target product profile and some insights into, the GMP manufacturing process for those of us who are thinking of, taking mRNA-based therapies through to the clinic. With that, we'll open up for questions for Lloyd. We have, 5 minutes, so we can take 3 or 4 questions, and then we'll move on to the second session.
Great. One of the questions, Lloyd, is whether lyophilization will help stabilize the particles at room temperature.
That remains to be seen. So I haven't seen any data recently, but I think that is something a lot of people are looking at. So I'd be very happy to, if someone has that data, to share. But again, I think we have to keep in mind all the work's that's been done right now with RNA and DNA vaccines is experimental. These are experimental vaccines. So I think initially the rush or the push is going to get an approved vaccine, and it's going to have cold chain. It's going to be stored frozen. So I think having a lyophilized or freeze-dried version of your vaccine is a longer-term goal and will likely replace the frozen vaccines that are being developed right now.
Yeah, if I can just help you there a little bit, Lloyd. You know, there are published data on lyophilization of mRNA on its own, and it's been shown to be, you know, thermostable for long periods of time, you know, at 2 to 8 degrees. And there's been plenty of work on, you know, freeze thaw of LNP mRNA vaccines, and that's possible. And there are people who've started to lyophilize these formulations. There's no published data yet, but, you know, there's common, you know, consensus that it's doable, but it's going to take, you know, a time and effort and resources before we're there. And certainly, you know, for now, proof of concept in phase 1, phase 2, we're seeing formulations going in, you know, frozen at -80. And we're going to see a lot more work happening over the coming years to really refine that target product profile for the future.
Right. So really just, you know, highlighting some of the challenges that we all face as a community working on this problem. Related to that is a question about the mechanisms of degradation for our mRNA LNPs. If you could expand on that.
Yeah, Andy, do you want to take a stab at that?
Yeah, sure. I mean, you know, the most obvious mechanism of degradation is hydrolysis. mRNA has a phosphodiester backbone, and in solution that will hydrolyze, and it's going to be concentration and pH dependent. And those mechanisms are quite well described. In addition, you're going to see oxidation and deamidation, and particularly there's you know, literature on the oxidation state of an mRNA actually results in disengagement from the ribosome and degradation of the mRNA. So both, you know, changed hydrolysis and clipping of the mRNA is important, but there are chemical changes in the mRNA that will impact the way that mRNA expresses protein. That's a little less defined, and typically, you know, you need to have a couple of ways of looking at your mRNA. You can look at it by HPLC and look for fragmentation. I would suggest that you also have some kind of biological potency assay where you put it on cells and you measure in a dose-dependent manner any changes in the ability of that mRNA to express its antigen in the context of a vaccine.
And we do know also, in addition to that, is that the lipid nanoparticle will protect the mRNA payload as well. So that's obviously one feature of the lipid nanoparticle.
Yeah, and that degradation is coming through RNAs. You know, the natural response of our body to viral RNA is to secrete RNAs. We breathe it out, we shed it from the skin, and certainly the lipid nanoparticle protects from that enzyme.
Yeah, and from the carrier, from the lipid nanoparticle point of view, from stability, we are seeing, or many people have seen, at least for sRNA, shelf life at 2 to 8 of much greater than a year. For frozen lipid nanoparticle formulations, the stability of the lipid nanoparticle is very good. So we're seeing upon freeze and thawing, those lipid nanoparticles maintain their physical chemical properties and maintain their biological activity.
Okay, thank you, Lloyd and Shell, for the questions. We're right on time, so I think it's time to transition to the second session, the breakout sessions, where people can go and sign up for one of the 4 sessions. So Jane, do you want to walk us through?
Yep, I'll walk you guys through. So what you see in front of you, Dana, will you go ahead and hit that link real quick? So what we're going to do now is we're going to move from one Zoom room to another Zoom room. And so we have 4 breakouts. We have breakout A, which was the basics of RNA and RNA vaccine. So that's the one that you heard about a little bit earlier. Then we have breakout B, which is Intro to RNA Delivery Technologies with Dr. Sehe. And then we have breakout C, which is the Antigen Design and Strategies with Annie Dosey. And then we have breakout D, which is Optimization of Lipid Nanoparticles for Intramuscular Administration of mRNA Vaccines. And that's going to be with Dr. Brito. And what we'll do is you're going to see that in your link, in your chat right now, you're going to see options for all 4. All you have to do is choose one of them. What you're going to do then is choose which one you want. You're going to click onto that link. It's going to immediately pop up to take you into that. But what it's going to do when you do that, your next thing you're going to see is some screens that might say one of these 2 things. One that might say you're going to be leaving the session. That's okay. Say yes, because that's just going to take you into your breakout. The other one that might happen if you have restrictions, kind of how you might be set up either personally or with your company, it may ask you to put your name and your email in again. Don't worry, you're not duplicating anything or anything like that. It's just more of something that's required in terms of how your computer's set up. And then it will ask you, are you okay to leave? Okay, we're going to be here. We're going to stay on here the whole time, so that we can walk you through this. If you have any problems or questions, you can chat us up, raise your hand or whatever, and we'll be able to keep an eye on it. But that's all you have to do. So now you just go in again to the chat, find which breakout you wanted to go to, click on that link, say yes, you're okay to go and leave this Zoom, which should bring you into the other Zoom. I don't know why this doesn't show up. There we go. Okay, we're going to put the links back into the chat. Okay, even if you just say, if you, maybe the way your computer set up, if you're seeing one that says, it says only copy URL. So if that's the case, you should be able to right click, and then it might, again, every computer is slightly different. So you should be able to right click on it and then be able to see it say, where it'll say open URL. You can click it that way. If you still can't get it to work, you can copy that and then just go into Chrome. Or whatever your browser is, put it in there and it should take you there. Just let us know if you run into any problems, but occasionally, you know, depending on how you're set up, it could be slightly different. And so we'll just keep an eye on it, keep working you guys through it till you get into your group. But like I said, the other option is you can right click and then it will say something like open URL. And then if you click that, that should get you there. Or like I said, you can copy it. I know we have a couple people who've raised their hands, so I'll try to get to you and you can ask any questions if you're running into any issues. Shell, can you do that for me? Can you go to the people that raise their hands and just open it and let them ask their questions? Because I think they're running into some issues. You just have to unmute them. Oh, can you, sorry, Dana, can you send the links again? Because new people just joined us and they just can't see it. So we're gonna add the links again so that you can see it if you just joined us, because you won't see the chat. So Dana's put it back in there. Again, all you have to do is go into the link. You can either, depending on how your computer's set up, you can just click onto it, or you might have to right click and then say go to URL, and it should take you there. It is going to give you a screen that's going to say, ask you a question about, are you okay to leave the Zoom? Say yes. It may give you a screen that asks you to put your name and your email in. That's just more because of the way your computer is set up, if you don't have cookies that remembered those things. So it will make you do that again. Don't worry, you're not duplicating anything or causing any problems, and you should be able to go forward, and you should be okay. Okay, so let's see how we're doing. Let's see how we're doing. We have almost everybody moving out, so I think we're doing better. Let's see. Shell, did we have anybody who has hands raised that needed to ask us a question? Everybody's good? Okay. Again, if you just joined us, just chat and let me know because we'll have to resend you the link. Oh, we do have somebody who's raised their hand. A couple of people. Hi, Bruno. Do you have a question you want to ask us? Okay, can you resend the link again, Dana? We're going to resend it in the chat. Again, your options are, you can see what your options are to go to. And then you can, and then I see we have somebody else. If you, I've unmuted you. If you wanted to ask a question real quick, I think we had somebody else that raised their hands. If you want to, you can ask me a question if you're running into any issues. Okay. All right, again, the chat, if you go to your chat, you'll see the links. If you just joined us, you won't see the link, so just let us know, and then we'll keep resending it. Just because when you come on new, if you hadn't been on there earlier, you won't have it. We keep this room open, so we can just help you get through this. I see we have a couple chats. Let me get over to here and see what we have. So, yeah, I think, can you do it one more time? Someone just joined us and she said she couldn't see it. Okay. So we'll just keep putting them in there so you guys can see them. So hopefully, Marjorie, you can see it now. I think that was Marjorie. Sorry, my eyes are not great. No, that was not Marjorie. Sorry, Mark. That was not your name. I was just looking at it quickly. But anyway, hopefully you can see it now and you can be able to click. Again, your options are you can either click directly onto the link, but based on your computer, you might have to do a right click on the link and then tell it to open URL if you're running into any issues. These are your options for your sessions, and then you just click onto the session that you want to attend. Bruno, did you want to ask us a question? You have your hand raised, and I've made it so that you could talk if you want to ask us a question, but you have to unmute yourself.
I want to go into breakout session A. Every time I click on it, I get into another session.
That's exactly what you want to do. So then you just hit yes, and it will take you straight to that session.
Yeah, but then when I click yes, I get into breakout session C or B. So I now tried 2 times.
Okay. So Dana, can we try just sending just session A only the link? Saying that when he's trying to do session A, he's getting other sessions. So can we just send just session A? So we're going to just put session A on the chat and no other session just to see if that helps you. So let's try that and see if that works for you, okay?
Thank you.
We'll keep it on just to we'll keep you unmuted just to make sure you're able to move over because then you'll just disappear if it works.
So I go into chat?
Yep, go into the chat. You'll see it should just show you A that just pops up right now. Try that one and see if that works.
Okay, so now I go launch meeting.
Yep.
And I open and I say yes.
Yes. It worked. He's gone. Okay, great. Okay. Okay, we'll do the same thing. Somebody's asked for session D. We'll do that one. Okay. I see we have a raised hand from Sahar. So I'm gonna unmute you if you wanted to ask us a question. So I'm allowing you to talk. So go ahead, Sahar. Do you have a question? Are you running into any issues?
Yes, please. I did the same thing when I clicked the breakout, I think B.
Okay.
No, yeah.
You want
For B,
session B? Okay, what we're gonna,
nothing happened.
okay, so did you try, okay, we're gonna put that link in there one more time, just only session B. Did you try doing a right click? Because it may be the way your computer's set up, do a right click and it'll say go to URL. Try that and see if something happens then.
Nothing.
Okay, the other option that you might try to do, it just maybe kind of something that's going on with your, the way your computer's configured, is to copy that URL, go into whatever you use, Chrome or whatever you use as your browser, just type that URL right at the very, you just copy and paste it and put it right there at your webs, where you go to a web, drop it in there and hit enter and see if that takes you there. Okay.
I did the same, nothing happened.
You did what? You went into the browser
Copy
and
and
tried
paste, yeah, and the browser, and then nothing happened. I don't know. And in the beginning, he said just join, leave the meeting, room meeting, and join another room.
Yeah, if it says that, you say yes.
Then it's disappear, and then I try again. Then I didn't find that blog for yes or no or something. Nothing happened.
Okay, so let's see here. Let's try one more thing. Okay. Sorry about that. My PowerPoint just dropped real quick. What's that?
You can use Zoom and join manually.
Okay, do you want to just tell her how to do that? You want to go on mute real quick? Shell's going to walk you through one way to do it because I just. Okay, go ahead, Shell.
So if you close Zoom, you can join basically any Zoom meeting with just the number. So you'll just have to enter the number manually. So take a note of what the number is for the breakout, and then you should be able to paste that into the window when you hit join in Zoom.
Can you send the links again? Okay, Dana has reposted them again. For those of you who just joined us, they are in the chat. So just to remind you the process, what you would do is you go in to your chat and then you're going to take the session that you want. You'll be able to go into it. You can either right click on the link and it'll say go to URL or you can click on it. When you do it, you're gonna see a screen that pops up that says leave session. That's okay. That's exactly the right way. It means you're going into the right, it's all working. Say yes. Then you may have, depending on what your restrictions are, you may have a screen that will ask you for your email and your name. Type that in really quick, and then you hit OK or submit, and then you'll be able to go straight into the session. So if you're running into an issue where you can't get the link to work at all because of some kind of setting you might have on your computer, you can copy and paste it and then go into the URL. I mean, go into the browser and put it in that way. And then Shell gave another recommendation where you can use the ID number and you can get into it that way. So hopefully we've got you guys getting, most of you are moving, but I still see we have a few that have not moved yet. So hopefully we can figure out what is happening. So there's 67 of you still in this room. That can be fine if that's just something you're doing because you're just, so don't feel like you have to. We're going to keep this room open, but if you are running into any issues, let us know. I see Michael Dosey, you have your hand up, so let me allow you to talk. Michael, are you having a problem and can we help you? Hi, Michael, you had your hand up. Michael Dosey. Okay, I am going to lower your hand since I haven't heard from you. And if you have any issues that you need, you can always go to us in chat. So if anyone else is having any challenges, just let us know. You can either chat with us or you can raise your hand and we'll get you into your room because I don't want you to miss out on anything. Some of the rooms are not recording because of the speaker's preference. So I just want to make sure you are able to get into any room that you wanted to get into. So just let us know if you're running into any issues. Otherwise, you're fine to stay here if you're just kind of using this as your, because you've got something else you're doing and you're just kind of staying into the session. That's fine. You can do that. I just don't want you not to be in a session you want to be in. Hi, if you just joined us, we have put the links into the chat now so that you can have access to them. Here's how you'll do it. You just go straight to the chat, click on the link. That is the link that you want to go to as to which breakout. If you're having any problems that doesn't go to the link directly, you can do a right click and it'll say go to URL. That should work for you. And then what we can also do, and then what will happen is you'll have a screen that pops up that will say leave Zoom. Yes, because when you leave it, it's just letting you leave this one and it's going to take you into the next one. So that's okay. Just go ahead and say leave Zoom and then go to the next one. And then that should get you into the right room. If depending on some of your computer restrictions, it may ask you for an email and a name. Just type it in real quick. You're not duplicating anything or causing any problems. That's just the way your computer needs it. And then just go ahead and that should take care of everything. If you're having any problems, go to chat, let us know, and we'll take care of you. Again, if you just joined us and you're wanting to look for a breakout, what you need to do is let us know if you just joined us because you may not see the links because you only can see the links in chat if you were already in the system. So if you need help, just let us know in a chat. Just say, I need to see the links and we'll get them to you. But basically in chat, you should be able to go there. If you've already been on, you can see the different links. You'll just click whichever link you want. You can either right click or go directly to it and we'll take care of you. Okay, I see Someone with the NS, that's what it says. You have raised your hand, so I will allow you to talk. So NS, if you raise your hand, are you having any questions or anything we can help you with?
Hi, I do not see the link, so can you please show
Yes.
or send it again in the chat?
Yeah, we're sending them again right now. Thank you. Let us know if you have any problems, but that should work.
Sure, thank you.
Again, just to remind everyone that you have the 4 breakouts right here. You go into the chat, you'll be able to pick out which breakout you want to go to. You'll see the link. You can right click or click onto the link depending on how your setup is. It'll pop up and ask you that you're okay to leave the Zoom session. Say yes, because you actually do want to leave the Zoom session. You may get a screen that will ask you for an email and a name. No problem. Just go ahead and type it in, and then you should get into your breakout. If you run into any issues, just let us know. Again, if anyone just joined us, just to remind you what we're doing now. Right now, we're in the breakout sessions. There are 4 breakouts. And then if you just joined us, you won't see the breakouts in the chat because you only can see the chats of when you were in a session. So if you just joined us, just give me a chat real quick. Just send a chat to say you need to see the breakouts, and I'll give you the links. Once you get the links, you just do a right click or just click onto it, depending on how your computer is set up. If it's a right click, you'll have a drop down and you'll say go to URL. And then it will say to you real quick that you're going to leave the Zoom. Say yes. And then from there, you'll be able to just go straight into the breakout room, or you may be asked for your email and your name. Just do that really quickly and it should take you there. Natasha, I have it set up so that you can talk. Are you having a question or you need some help?
Hi, no, sorry, I just logged into a different computer.
Okay, okay. So do you need to see the links again or are you okay?
Yeah, do you mind sharing the links again, Jane?
Yeah, no problem. We'll do that.
Thank you. Sorry to cause
No, no,
problems.
no problem. Okay, bye-bye. Oh, okay. Can you put the links in again one more time? Okay, thank you. Just to give you just a quick heads up, what we're doing right now, we are in breakouts right now. And we'll be wrapping those up at the top of the hour. So we only have about 10 more minutes in breakouts. But if you wanted to attend a breakout, you see what the sessions are. If you go into chat, you can see the links. If you just joined us, you may not be able to see the links and you just need to let us know and we can resend them. But if you are waiting just for the next session, you're in the right space. You don't have to leave. You can just hang out. In about 10, 12 minutes, we'll start back up with everybody coming back together. So after the breakouts, everybody will be rejoining us in this session. So you don't need to leave if you just want to hang out for about 12 minutes. And then we'll be started up with our session number 3. So you're in the right spot unless you want to go to a breakout. Otherwise, you're fine just to hang out with us.
Happy to hang out, Jane.
joined us. We only are just a few minutes away. We're just wrapping up our breakout session. So we're only just about 5 to 10 minutes to give everybody a little time to move over. So we have about 5 to 10 minutes and then we'll be moving over. So you're fine where you are, you're in the right spot, just hang out with us, and then we'll be starting in about 5 to 10 minutes as we wrap up the breakout sessions, and we'll start our next session.
Jane, can you hear me?
And we can. I sent you a chat under the panelists if you could look at it real quick.
Okay. Yes.
Hi everyone, thanks for joining us. So in just a little bit, our breakout sessions will be ending, and we'll be having you all move in. People will be starting, so they're already starting to happen. They're starting to move in. So just give us a few minutes to get all of our breakout sessions closed out, and we're getting everybody back into this main session. So just give us about probably going to do about 4 minutes, 4 to 5 minutes, if you don't mind, just kind of hanging out for a minute. Great time to grab that cup of coffee, take a quick break. But we should be starting in about 4-5 minutes as we get everybody moved over back into this general session. You don't have any more moving to do, so thank you for working with us virtually to get to your breakouts. And then don't worry, you're in the right session for the rest of our time. So once you get here, I won't move you anymore. Again, if you just joined us or moved over from a breakout room, you're in the right spot. We're going to be starting in just a few minutes, just giving everybody opportunity to get moved over from their breakouts. Thank you very much for hanging in there with us, and we'll be starting in just a few minutes. Hi, thanks for joining us. Welcome back if you're coming in from a breakout. We're going to be starting in just a few minutes. You're in the right spot. We're just getting everybody moved over from the breakouts. As I said earlier, we're not moving you anymore, so thank you for working with us in our virtual world and going from one to the other, but we're seeing our numbers coming back up, so we know everybody's getting moved over. We're just going to give everybody a few minutes to get moved over, and then we'll start right back up. Again, thank you all for joining us. We almost had everybody moved over, so we're going to start, just to give you a heads up. We'll start in 1 minute. So Andy, just to give you a warning, we're going to start in 1 minute.
Okay, I'm ready, Jane. Thanks.
Okay, Andy, we're ready to start whenever you are.
Okay, Jane, I'm ready. Let's put up the slides.
We can't hear you if you're talking, Andy.
Can you hear me now? Can you hear me now, Jane? Can you hear me now?
Still can't hear you.
Can you hear me?
I can hear you, Andy.
Okay, we can.
Okay, so can we go with the first slide?
Okay, Andy, are you talking?
I am now, yes.
Okay, good, got you. Yep, you're good. Okay, right, great. We're ready to start.
Okay, first slide. Thank you. Okay, everyone, welcome back. We're now in the demonstration part of the symposium. Here we're going to talk about the fourth pillar of mRNA vaccines manufacture, and we're going to focus on formulation of that non-viral delivery system, and we're going to focus on an LNP. So next slide. So back in 2012, the group that I led at Novartis Vaccines were actually the first to publish on the use of a lipid nanoparticle to deliver an mRNA vaccine. And we were using at the time self-amplifying mRNA. And we did our pivotal studies in RSV, where we were expressing the F glycoprotein from RSV. And we went ahead and we made vaccines with an ionizable cationic lipid that had been published by a group at TechMira. And in fact, it was Lloyd, who I now work with at PNI, who was on that paper. They described the structure of the cationic lipid and then how to make those lipid nanoparticles using a process of ethanol dilution. And so we used that published literature to make these formulations. And we showed for the first time that mRNA vaccines were very potent in mouse models and capable of eliciting immune responses. And they were equivalent to the same RNA delivered with a viral replicant particle produced using a packaging cell line. So how did we go about making those formulations? This biology was published in PNAS, but we followed up with a paper, if we go to the next slide, Jane, where we described the manufacturing process. So the lipid nanoparticle we were using was 4 components. We had a neutral lipid DSPC forming the base of the particle, cholesterol helping to stabilize that structure. The cationic lipid was an ionizable cationic lipid, DLIM-DMA, and we included PEG lipid in there to stabilize the lipid nanoparticle, particularly during the downstream processing for tangential flow filtration to remove the ethanol, do buffer exchange and concentrate, and also for sterile filtration. Next slide. Morphologically, these particles by cryo-TEM were quite diverse in structure. We had, multilamellar liposomes. We had all sorts of different structures, sizes varying from like 100 nanometers up to 140 nanometers. But we went ahead, we vaccinated mice, and the formulation worked. Next slide. This is the setup that we used. It's quite complex, and it took a lot of time to set up and make the formulations. We had a tea mixer, 3 syringes on a syringe driver, and we drove the ethanol solution with lipid and the RNA through a tea mixer, and then diluted after mixing into a container, and then sterile filtered that material, went through tangential flow filtration. At the time, Spectrum Labs had developed a small scale TFF system that was capable of processing around 2 mils of formulation. And so we were able to mimic an industrial process, inline mixing, TFF, sterile filtration, and those formulations went on into the preclinical models and showed that they were very successful. But this was not seen as a scalable process at the time, given the difficulties of setting this apparatus up. And it took us a long time in the lab to run iterations of formulations and vectors to end up with a final product that we were happy with. And so this technology never really moved forward. Obviously, 2012, this was all published, and a follow-up paper in 2015. But what has changed? A lot's changed, and now we've got a chance to see what P&I has done in terms of innovating with microfluidics to make this process much more amenable in the lab, both in the preclinical setting and also transitioning through into GMP. So with that, I think we're going to show you the first video and a demonstration of the Ignite system, which is the benchtop system that you can use in the preclinical setting, 1 to 15 mils, ideal for optimizing your mRNA vaccine.
The NanoAssembler Ignite is a powerful tool for advancing your nanomedicine development. We took our next-gen technology, which was designed for GMP production, scaled it down, and designed a system around it to enable scientists of any skill level to explore the nanomedicine design space. In this demo, we'll be making enough lipid nanoparticles to encapsulate all the mRNA needed to meet the dosing described in the Gale 2012 paper. Based on the N/P ratios used, this can be achieved in a single 5 milliliter Ignite formulation. The Ignite's interface puts key parameters that define the conditions of particle formation, such as the total flow rate and the flow rate ratio, literally at your fingertips. It ensures that conditions are highly reproducible, run after run, regardless of who's using it. With Ignite, NextGen has been scaled down and engineered into a format appropriate for preclinical development work. Fluid input is handled through syringes which attach by Luer lock directly to the Ignite cartridge. Here, RNA in an acidic buffer is loaded in a syringe. Then our Genvoy ionizable lipid mix dissolved in ethanol is loaded in another. The Ignite will inject these into the NextGen mixer under the conditions specified to control the nanoprecipitation reaction. Because the same NextGen technology is available across the NanoAssembler platform, users are able to work with and start developing a process representative of the eventual GMP process. So you're not only making formulations for preclinical studies, you're also getting a head start developing that final process at the earliest stages. The ease of use of the Ignite addresses another appreciable challenge faced by the field, namely assembling or training a workforce skilled in the art of making LNPs. The Ignite lowers the barriers to successfully formulating nanomedicines and reduces human error. This means you don't need to spend years in the lab perfecting a technique. Instead, new users can get up to speed very quickly, parameters can be transferred between users or to other departments or between collaborators, so researchers can concentrate their efforts on the multitude of other activities that are crucial to developing a vaccine or other nanomedicine. At the heart of the Ignite is this NextGen microfluidic mixer. We created NextGen to address the challenges of clinical and commercial scale manufacturing. On a GMP system, a single mixer throughput of over 200 milliliters per minute is possible, while maintaining the control and reproducibility that is the hallmark of NanoAssembler non-turbulent mixing technology. The fluid path is completely disposable, mimicking what you would do in the GMP environment. This also reduces the risk of cross-contamination and eliminates the priming and cleaning steps, which greatly streamlines the workflow. Here you can see just how fast Ignite produces this formulation. You'll also see Ignite automatically divert a small portion of the formulation at the head and tail of the process to ensure only the best quality particles produced under steady state conditions are collected. And here you have a 5 ml formulation which encapsulates more than enough RNA to meet all the in vivo dosing requirements of the paper. You can make formulations between 1 and 15 ml pre-dilution, which is ideal for exploring the formulation design space and evaluating formulations in a range of in vitro and in vivo models. The next step for this formulation is to prepare the particles for dosing. This involves particle purification by either centrifugal or tangential flow filtration, and finally sterile filtration. The ease of use, speed, control, reproducibility, and scalability make the Ignite a tremendously powerful preclinical development system.
Okay, so I think we've seen, a great demonstration of the Ignite, the benchtop system, ideal for that, formulation optimization, screening, different iterations of antigens and vectors working in the 1 to 15 mil range. But we also need to keep an eye on that you need a process that's scalable if you want to go to the clinic, and there are some downstream unit operations that you need to consider. You've got tangential flow filtration or dialysis to remove ethanol, exchange buffer, concentrate, and then you've got sterile filtration. So one of the benefits of that microfluidic system, if we move to the next slide, please, Jane, is exquisite control of particle size. Here we've just got data demonstrating 2 different mixing scenarios. In the orange, we've got microfluidic, and in the gray, we've got inline mixing. And what you can see is a much tighter particle distribution and a reduced polydispersity. If we're doing sterile filtration, we've got a molecular size cutoff of 200 nanometers. And if you've got bulk solution, you're trying to sterile filter. Eventually, if you have particles around and above that 200 nanometer, you're going to bulk the filter. So microfluidics helps you through that scale-up process and that bulk filtration. So if we go to the next slide, please, Jane. Also, because we can scale with the next generation chip all the way through the different platforms to GMP and the high capacity system, we're able to maintain particle morphology. Using the microfluidic system, we tend to see this solid core morphology, and it's maintained throughout the different platforms. In macro mixing, you tend to see multi-lamellar morphologies, and these morphologies can change during the scale-up process, and this could have an impact on biological activity. So if we go to the next slide, please, Jane. So here's the platform that we offer at Precision Nanosystems. We've shown you the Ignite. The next step up is the Blaze. This you can formulate up to a liter of concentrated formulation before dilution. It's ideal for those preclinical tox studies and when you're starting to think about scale up and moving towards clinical applications. So with that, I think we have the next gen Blaze video.
The next-gen Blaze is designed to take formulations developed on the Ignite and rapidly scale them up for late preclinical development. This supports larger scale in vivo tox studies in multiple species and process development to scale upstream and downstream operations to rapidly transition to the clinic. Blaze is enabled with our next-gen technology. We've made next-gen available across the NanoAssembler platform to allow you to scale your formulations across any stage of drug development. An intuitive software interface allows you to easily dial in parameters like total flow rate and flow rate ratio. The Blaze offers the choice of 2 next-gen mixers and operates up to 90 mils per minute pre-dilution. You can easily scale your bench formulations up to 1 liter or combine 2 unit operations in one using inline dilution to make up to 5 liters of bulk nanoparticles. Such a formulation requires just over 10 minutes of formulation time, and using typical formulation parameters is enough to encapsulate 260 milligrams of mRNA. You can choose between Next-Gen 400 or Next-Gen 500 mixers, the latter being identical to what's used in the GMP system, so you're able to develop your scale-up process with the same mixer you'll eventually use for clinical manufacture. Here we are making 40 mils of undiluted formulation using Next-Gen 400 at 60 mils per minute. The The run takes about 40 seconds and formulates 10.5 milligrams of mRNA in lipid nanoparticles. Following this, a dilution step is generally employed to prepare the sample for downstream processing. Together, NextGen and Blaze greatly simplify scale-up of the unit operations for both particle production and inline dilution. So you can focus on scaling upstream and downstream processes and building that data package to support regulatory applications. Ultimately, the Blaze helps to minimize risk, timelines, and cost of late preclinical and scale-up development.
Okay, so there we had a nice demonstration of the Blaze system. So at the heart of the PNI's nanoassembled platform is that next generation chip. The microfluidic engineers have redesigned that herringbone system. So we now have a next generation fluid channel that we can make geometry changes in. And so on the graph, we're showing how the geometry is changing from extra small to large, allowing increases in flow rate, allowing us to scale. We maintain particle size and morphology during this process, but allowing you to scale from that benchtop system, the Ignite, all the way through to the GMP system. In this case, the target size limit was 40 nanometers, and you can see with the large channel next gen chip, we're getting flow rates up above 100 mils a minute. And so now we're going to show you, a demonstration video of the GMP system, which has just been released.
The GMP system is one part of a full service solution we call Clinical Solutions that helps you manufacture your lead candidate for the clinic and beyond. Having worked with nanomedicines for over 20 years, including many clinical stage projects, I understand firsthand many of the challenges in clinical manufacturing. Our dedicated team of formulation and analytical scientists, process and instrumentation engineers, supported by our quality group, are ready to solve unique project challenges. The NanoAssembler GMP system has been designed to streamline nanomedicine manufacturing in a GMP environment. At the core of the GMP system is the same next-gen microfluidic mixer that is available on the Blaze, allowing smooth transfer of late preclinical formulations onto the GMP system. The fluid path is designed to be single-use and easily replaceable, avoiding costly and time-consuming cleaning validation. The entire fluid path has full materials traceability to facilitate compliance. The fluid path is supplied double-bagged to avoid contamination, and the microfluidic mixer is gamma irradiated to ensure an efficient workflow. To support the technology transfer and operation of the GMP system, we provide a complete documentation package including SOPs and protocols. If needed, our clinical solutions team is ready to support customization of the GMP system to meet your process requirements. The interface is simply controlled through a tablet PC, so like its nanoassembler siblings, it's intuitive to dial in precise formulation parameters and is easy to operate. With this standard configuration, we have produced challenging formulations at particle formation rates of 12 liters per hour and can produce nanoparticles at even higher flow rates. The standard GMP system includes an inline dilution pump that prepares the sample for downstream processing, such as tangential flow filtration. In this demonstration, we are performing a 30-minute run at 12 liters per hour to produce 6 liters of pre-diluted formulation. After inline dilution, we are collecting 24 liters of formulation into 2 bioprocessing bags. As with other nanosampler systems, the GMP system has an important sample switching feature to allow collection of formulation when the system has reached steady state flow. Our team has repeatedly demonstrated that formulation results on the blades are replicated when moving to the GMP system. P&I is now transferring the GMP system to multiple CMOs and client GMP facilities around the world. P&I has now enabled our client, Cytonomics, to successfully produce a GMP batch using a customized system. P&I's technology made it possible to accelerate scale-up, technology transfer, and GMP manufacturing to be completed within 6 months. Once nanoparticle formation is complete, we have finished 2 of the unit operations necessary for a GMP nanomedicine batch. Following sample dilution, the in-process batch size is now 24 liters. This volume is equivalent to formulating approximately 1 gram of messenger RNA based upon published N/P ratios and input concentrations. Precision Nanosystems GMP system produces nanomedicine batches for clinical use. The seamless scalability of our technology accelerates the development of promising nanomedicine candidates from the bench to the clinic.
So we've shown you, videos of the 3 nanoassembler technologies that P&I provides. I think it's important to demonstrate that, the scalability across the platform. So here we're just showing you an LNP mRNA formulation. The mRNA encodes EPO. We're just showing across the different devices, Ignite, Blaze through to GMP, with the different flow rates, with the different chip geometries, we're able to maintain particle size and the polydispersia of those sizes here on the left-hand side. And on the right-hand side, we're just measuring the encapsulation efficiency. And again, showing across the different devices, the different chip geometries, we're able to maintain all of those chemico-physical properties of the particles. If we go to the next slide, it's also important to demonstrate biological activity. EPO is expressed in the liver, so these formulations were administered intravenously to mice, and then we went ahead and measured the total serum expression of the recombinant human EPO. And what you can see across the different platform, either using spin columns or tangential flow filtration, we were able to maintain the expression levels of EPO from the liver in the mouse. And on the right hand side, EPO will have an impact on the hematocrit, the amount of red blood cells in the blood. And what you can see is a consistent response and increase in hematocrit in the mice across the platform, from Ignite through to the GMP system and at the different total flow rates. The GMP system, obviously at high capacity, 200 mils a minute. So with that, I'm going to hand you over to Angel, who's going to talk you through, how do you, get in contact with P&I and start talking about getting access to our platform technology.
Thank you, Andy. Appreciate the intro. Good morning, good afternoon, and good evening. My name is Angel Galarza. I'm the Director of Sales for the West here in the United States of America. Here at Precision Nanosystems, we have a support system for all of our customers and those interested in learning more about our technology throughout the world, either via direct support or through our distributor network, our local distributor network. I'd like to take some time to introduce our teams. Here in the North America West, we have in the Southwest region, we have Kim Killian and Viet Nguyen, our application scientist, and our regional sales manager. In the Northwest, we have Ian Villamagna and myself. If we go to the next slide. In North America East, we have Todd Quitchoff and Sri Kakumanu, as well as Jason Coleman and Pratik Goswami. They're based out of our Eastern region. Out of Europe, we have our local support team, AJ Johnny and Jürgen Schmidt, as well as Richard Broadhead, our field application scientist, and Martin Rabel. They're in the European region. In our APAC region, we have Mal Lau and Anastasia Darwatan, as well as our local distributors in the different areas. If you have any questions, feel free to reach out to our local representatives, and we'd be happy to answer any of your questions that you may have on our technology. Thank you very much.
Okay, thank you. And so we're going to move into our fourth session, the panel discussion, give you all an opportunity to ask questions. I believe in the mRNA session, we ran out of time, and there's been a request for Jessica Madigan to present some slides and thoughts on enabling GMP production of mRNA. So we'll work to get those slides available. So Jessica, if you could send them to Jane. In addition, I just wanted to, there was one person on the panel who hasn't participated in any of the discussions. That would be Suraj Abrahams. So he's from PNI. He's in the formulations group, but he's also setting up a molecular biology lab at PNI. We're focused on generating self-amplifying mRNA vectors. So, if there's any questions on self-amplifying RNA vectors or, PNI's approach to LNPs, he or Lloyd can answer those questions. So with that, I'll open up for questions. Hopefully we can get Jessica's slides available, and when we're ready, we can, give her an opportunity to present those to everybody. Over to you, Shal. I believe you're going to bring in the questions.
If we can ask all the panelists to show yourself so that we can bring everybody up. And I think we've got Lloyd, we've got Jessica, we have Annie. So everybody, if you're on there, if you could just show yourself, we will open up for questions. Here we go. We're starting to see everybody. And then Jessica, what we want to do, if you want to, are you at a place now that you could show your stuff? Okay, why don't we let Jessica, you guys stay, you're fine, but why don't we just go ahead and let Jessica go ahead, and you can share your screen and do your presentation, because big part of what we're going to talk about now is manufacturing. So it's a great segue to then open it up to the questions that you guys all have.
All right, can everyone see this here?
You're perfect.
Great. Thanks so much for making some time for us to finish this presentation today. And I also just want to thank Anton for covering the important considerations for mRNA manufacturing prior to me jumping in. So for me, I'm going to take the next couple minutes to cover additional considerations for GMP manufacturing, including timelines, facilities, quality control, specifications, and stability. So what I'm showing here is pretty much what we consider kind of a hair on fire timeline. This timeline is an actual representation. Oops, sorry, don't know if I misshared there, but is an actual representation of what we were able to accomplish for the development of a SARS-CoV-2 vaccine that is currently in clinical trials. If you add up all the days here, it totals about 63 days. And so this is extremely accelerated. Average timelines for standard clinical development of an mRNA manufacturing is approximately 9 months. So what
Jessica, I'm just going to interrupt you real quick because your screens are cut off a little bit. Can you just go to full screen? And then I think we'll be able to see your so to the top, there we go. Perfect. Thank you.
You can see the whole thing now?
Yep, now we're good. Thank you.
Perfect, okay. So you're able to basically leverage TriLink's platform manufacturing process for accelerating these clinical timelines. In the next slide here, I am showing our new manufacturing facility, which we moved into in 2020. This manufacturing facility is basically our commitment to growing with our clients through the manufacturing phases. We have about 105,000 square feet of facility. And within that facility, there are 5 manufacturing suites for GMP manufacturing. So we have the ability to do parallel manufacturing of 5 different clinical programs at any given time. Here I'm showing the traditional stages of therapeutic development pathway. Research grade material is typically used for toxin preclinical production, and GMP material is used for phase 1. One option to condense timelines for a clinical program is to produce a single batch as tox and phase 1 under GMP conditions. This would be considered a bit of a risk to the program because you wouldn't have relevant animal data to know that the material will be effective, but it definitely will help you accelerate timelines. And so if you want to take that risk-based approach, TriLink is here to help assist with that. After successful phase 1, we would move to a scale-up process where we're going to be making sure that the manufacturing process is achieving the amount of material needed for later phase production. And once that process development has been completed, you would move again to GMP production for later phase materials. When selecting a CMO, it's important to choose one that can meet all of your quality needs throughout the different phases of your program. So TriLink has the ability to manufacture basic research, diagnostic, and preclinical and clinical materials. But everything that we manufacture here, even for your research grade syntheses, is fully traceable and documented under ISO 9001 conditions. And our GMP facility is in compliance with ICH Q7 Section 19. A lot of the times when considering a new therapeutic development, the question of what grade of material do you need comes up. And you need to consider what grade of material not only do you need to execute your clinical trial, but also the raw materials that enter that process. So if you read the guidance from regulatory agencies, it does state that GMP grade materials should be used to make GMP grade product. But oftentimes GMP grade materials can be cost prohibitive or timeline prohibitive depending on what the scope of your scale or your program is. So you will need to do kind of a risk-based approach, and we encourage our clients to interface early with regulatory agencies and pre-IND meetings in order to establish what quality of raw materials is going to be suitable for their program, especially with respect to enzymes and plasmid. So now after you've kind of developed your manufacturing process, you need to start considering which assays need to be used in order to release your product for phase 1. By referencing the guidances, you'll see that you'll need to have ID testing, strength testing, and impurity testing. So we're always encouraging our clients to choose a robust panel of assays. At TriLink, we offer a number of different options for purity, including fragment analyzer, and bioanalyzer options, and HPLC options. For safety testing, you're going to want to include endotoxin and bioburden. And then you're going to want to look at process-related impurities, such as residual DNA and residual protein, and potentially residual solvents, depending on what type of manufacturing method you're using. After you've selected the assays, you're going to want to set specifications. The important thing here is to make sure that your specifications are achievable, and they're going to be dependent on the stage of development. So we encourage our clients in early phase to make sure that the specifications are as broad as possible so that as you advance through later phases, you can begin to tighten those specifications. And ultimately, it is the customer's decision to design what specifications are going to be met for likelihood of regulatory acceptance, but we're going to guide you on what's achievable. So for example, for a self-replicating messenger RNA that's going to be greater than 10,000 bases, setting a purity of greater than 90% by CE is not going to be achievable. So you need to make sure that you have efficacy data that demonstrates what purity is achievable and set the specs accordingly. And then it's important to note that final release and retention for GMP is actually probably going to exceed the amount you need for your study, especially for phase 1. So you need to consider that when setting manufacturing scales. After you've set specifications and decided which assays you're using, then you must make sure that the assays are fit for use. Methods should be qualified, and so you're going to want to look at a range of options, including accuracy, precision, specificity, limit of detection, limit of quantification, linearity, and range. And important to include in your timeline is that method qualification can take a bit of time. Average timelines are 6 to 12 weeks. So you want to build that into your considerations. And finally, I want to remind everyone to not forget that in order to file for your IND, you must submit at least 1 month of stability. For a messenger RNA program, you're going to want to consider long-term storage conditions as well as accelerated, and you're going to want to choose a panel of assays that are going to be stability indicating. Then you're going to choose time points, at minimum out to 12 months. Oftentimes we encourage our clients to go to 24 or 36 months, depending on the length of their trial. But we do offer a variety of options, and we're here to happy to discuss your program to find the best fit for you. So I will end there and thank everyone for their attention and look forward to the discussion panel.
Okay, thank you, Jessica.
Okay, all of our panelists, good, you're all there? Okay, you guys are ready for Q&A.
Okay, so Jane, this is the biggest challenge that we face, not all being together, not getting visual cues. So I think Shell's gonna, ask the questions. If panelists wanna, chime in, if it goes silent, I may answer the question or I'll try and divert it to the appropriate person. So with that, let's begin, Shell.
Sure, let me start with a question that was residual from one of Lloyd's early presentation there. Someone was asking if you could elaborate on how you can decentralize manufacturing and how long it would take to set up such a facility and what kind of production volumes can be expected.
Yeah, well, I'll start off and then maybe Andy can add. What we're thinking is, the GMP system that was shown in the video a few minutes ago, all the process steps fit within a pretty small footprint. So you don't need a very big space to do that. And we are currently with the GMP system and also the downstream systems as well, provided by companies like Cytiva, are available throughout the world. So the idea would be is to have a portable system, a complete solution that can be basically installed anywhere where there's a clean room. So that would be the first option. The second, maybe longer term option is a complete solution where you'd have a self-contained facility that could fit on the back of a truck or in an airplane and could be dropped anywhere in the world.
Yeah, so if we can just build on that, the concept is being explored by various, funding agencies. We've seen announcements from DARPA for, mobile manufacturing of an RNA vaccine. Conceptually, this would look like, 10,000 square foot pod system that you could put in an existing warehouse. Transcription reaction could run from, 1 to 10 liters. The formulation process would then scale up, up to about 100 liters. Capacity would be dependent on the type of mRNA and the dose of the vaccine required. Self-amplifying RNA, much lower doses. As we move through to the base modified, you're starting to look at tens to hundreds of micrograms. The capacity drops, but as a platform technology, those units should be capable of supplying millions of doses. The bottleneck would be in the fill finish and that final composition in the vial. Hopefully that helps answer the question.
Great. So for our next question here, I was wondering if perhaps the panel could weigh in on one of the questions we've had is We've spoken about capacity in terms of volume, but volume is obviously, how much volume you need is dependent on many things, such as your concentration of your API and its potency and all of these other factors. So I wonder if you guys could weigh in a little bit on how you bring all these things together to start to, at what point do you start to look at increasing your concentrations at the same time as looking at your potency, and how do you bring all of that together to estimate how much you'll need for your initial trials in GMP production?
So Jessica, do you want to take a stab at that from the mRNA side, and then maybe we'll have Lloyd on the formulation side?
Absolutely. So, from TrilInk's perspective, I think that, scalability approach to this is pretty simplistic. Messenger RNA manufacturing takes very little footprint. The reactors available that have already been used for protein manufacturing can be easily ported to messenger RNA. We can go, I think there's up to 50-liter reactions already available. And so it's just really looking at, meeting that criteria. And, to your point, the concentration of RNA that you need will obviously drive the volume of reaction that you need. Additionally, you're going to want to look at the stability of the product and how quickly you're going to turn around delivery and distribution. So there's a lot of factors there. But at TrilInk, we're easily comfortable in multi-liter reactions at this time. And these reactors come in larger sizes already from the manufacturing people for those pieces of equipment. So
Jessica, how much would you say you would lose of your material for in-process testing, those stability studies? Is it a third? Is it just a ballpark for everybody?
Yeah, so obviously it's going to depend on your starting scale. At minimum, when we're talking about a phase 1, we're probably dedicating about 100 milligrams of product to QC and to stability testing. And so that doesn't sound like a lot, but for a phase 1, if you're only using 10 milligrams to, 100 patients, especially for a self-amplifying type of product, that's 10 times the amount that you need. But once you're in later phases and you're making multi-grams, 50 mgs is nothing, 100 mgs is nothing. So it's all pretty relative. Early phase, that makes a huge impact. As you get to later phase, I think obviously that tails off a little bit.
Okay, thank you. And so Lloyd, what about, volumes, concentrations in the formulation process? Is there some range and variability there?
Yeah, so to carry on from Jessica's comments, for a phase 1 drug product yields and for the amount required for quality control testing, for QA retains, is proportionally quite high, given typically the smaller batch sizes for RNA LNP drug product. However, as you scale further, we're seeing yields, total yields in excess of 80%, even after QC sampling. So that's quite impressive. At the same time, as you scale, I think a key consideration is you scale the equipment as well, so that the compounding of an LNP drug product should take place with a single day. It might be 2 shifts in your clean room, but between 8 and 12 hours, you should be able to complete the processing steps from actually taking your RNA and mixing it with your lipid solution all the way to a bulk that's ready for a bioburden reduction filtration that would then be followed by a sterile filtration and then filling into your chosen presentation. So for downstream, those scale-up systems exist. And for the particle formation with the nanoassembler GMP system, with the current demonstration we're showing you, we're formulating 1 gram in about 30 minutes. And we can basically scale that all the way up to a 5 or 10 gram batch right now. Having parallel systems or parallel mixers, we can scale that even further. But given the highly potent nature of many of these mRNA drug substances or APIs, larger scales really aren't that required like they would be for, say, a small molecule drug product.
So we'll just open it up to the rest of the panel, see if anyone wants to chime in. If we hear silence, we'll go to the next question. Next question.
Okay, so I'll try and segue that topic into a somewhat related one. I think Jessica touched on it somewhat when she referred to sort of the potency and the link that has to the scale that you're going to need. But related to potency might be some of the effects like anti-vector immunity, and there were several questions about that. I know, Andy, you probably have something to say about different delivery methods and anti-vector immunity, but there were questions about whether or not we're getting immunity against LNPs and what effect that has. That there's limiting the number of doses you can have, et cetera.
Yeah, I can certainly lead off with some answers there. There is PEG on the LNP. It's a short chain PEG, usually molecular weight 2000. Those antibodies, if they exist, are pretty low binding. Transfection happens relatively fast. In the preclinical models, there's really been no evidence of any detrimental effects that you might see to those antibodies if they exist. So not inhibiting the platform. This is unlike viral vectors where you have a protein shell and the immune system, if it's seen it before, is capable of mounting an immune response. And so if, for instance, CanSino is in the clinic with an Ad5 vector for COVID-19, they see pre-existing antibody titers, they're only able to dose once and they have to dose quite high. If you look at the data from Oxford, they have a chimp adeno system where there really isn't pre-existing immunity, and they're able to dose twice, but they do start to see anti-vector immunity. The antibodies for PEG are much lower affinity, and so there's no evidence. One of the arguments against self-amplifying RNA is it expresses non-structural proteins to allow amplification. There's no evidence both for replicons delivered with viral systems and with lipid nanoparticles that any immune responses to those non-structurals has a detrimental effect on the vaccine as a platform with subsequent vaccinations. I don't know if there's any other comments from the panel.
Okay, then I guess we can go to a live question now. Jane, I think you're unmuted. Please go ahead.
Hi, yes. One question that I had was regarding the protein expression following naked mRNA injection. And one thing that several panelists have discussed today is that injection of naked mRNA can result in robust protein expression, but doesn't always result in a robust immune response following intramuscular injection. So why exactly does this material need to be formulated into an LNP or some other delivery vehicle to actually achieve robust immune response, despite the fact that you are able to achieve protein expression following naked mRNA injection?
Well, I'll give you some data that we generated at Novartis in a vaccine setting with a self-amplifying RNA. So we did some mechanism of action studies and published those data. So the vaccine worked two ways. We had the initial intramuscular injection. We transfected a few muscle cells. That RNA launched in the cell, expressed the antigen in the cell, and began self-amplifying. A lot of the vaccine was picked up immediately by the immune compartment, antigen-presenting cells, for instance. It didn't result in antigen expression, but resulted in an adjuvant response. The RNA hit the pattern recognition systems within the immune compartment. You got a type 1 interferon, and a danger signal. The immune system came in at the site of injection and found the antigen. There was a later stage of antiviral response as the RNA amplified in the cell. About day 7, cell went into apoptosis, and the cell was cleared by the immune compartment. So quite a complex mechanism of action. Antigen expression lasted for about 7 days. If we put a non-immunogenic protein in, like luciferase, it lasted for months. The moment we put an antigen in, it truncated very quickly. And so an important part of the vaccine mechanism of action is the RNA inside the LNP traffics to the immune compartment and adjuvants the immune system, and this will impact how the RNA expresses the antigen. And overall, for the self-amplifying systems, protein expression driving protein expression didn't increase potency. Changing the way the LNP engaged in the immune compartment did. And so it's for that particular type of vaccine, that seemed to be the dominant effect. LNPs were capable of adjuvanting the vaccine in an antigen-specific manner. So any other comments from the rest of the panel?
Perhaps we can move on to the next question then. And we've got a question here. We've talked about expressing antigens in a vaccine through mRNA, but could an mRNA vaccine be used to express an antibody?
So the answer to that is absolutely yes. So you can express antibodies from messenger RNAs, and that is a viable way. I don't know that I would necessarily call that a vaccine, strictly speaking, because it's not an antibody generated by the immune system, but it would be a prophylactic antibody, perhaps.
Yeah, just to kind of piggyback on that, a lot of people's approach to antibodies actually is to encode for 2 separate messenger RNAs, one for the heavy chain and one for the light chain, and then mixing those together to deliver a therapeutic.
Maybe Suraj or Lloyd could comment about the different types of delivery system for vaccines versus this kind of gene therapy approach.
Yes, with respect to the, I think most of the NEB and trialing could watch on me also in terms of like there are some bicistronic approaches where we design the vectors with a bicistronic, like using a heavy chain or light chain antibody. You can make mRNAs and you can express the antibody. But there are lots of self-assembling proteins that has been inserted into these vectors, and these are then introduced into the design that you want to make this DNA templates that you use to make mRNAs, and you can work around to either make a self-amplifying or a conventional mRNA. So it is a possibility to do that, and there are tons of literatures around it showing different designs. But whether that has been translated into clinical use, it's still questionable because it's still in the research phase.
Good. And another consideration is obviously when you're designing the lipid nanoparticle carrier for your RNA, you have to keep in mind the destination where you need that RNA to go and what cells or what tissue types that needs to be expressed in. So that's an important consideration for IV administration and also obviously for local administration as well. So a formulation that would be good, let's say IV administration targeting the liver, may not be optimal for a vaccine application or a tumor delivery or oncology application.
Great, thanks, Lloyd. And then I think somewhat related to the original question, there was a question about combining, say, an mRNA encoding the antigen with an siRNA that knocks down some factor in the virus itself, or some cytokine stimulating type things. If you knock that down, can you combine that to make a treatment and a vaccine in one?
From a manufacturing standpoint, we know that for the lipid nanoparticle, you can encapsulate multiple different RNA species, whether it's multiple siRNAs multiple mRNAs. The actual delivery system is somewhat agnostic to the RNA that's encapsulated. So that's the manufacturing kind of response or answer to that question. With respect to preclinical, I might defer that to someone else.
I guess I can comment, having worked on antiviral siRNAs in the past. The challenge with doing antivirals with siRNA is really your transfection efficiency and what percentage of the cells that are infected by a virus you can reach with the lipid nanoparticle. And so that's a relatively challenging endeavor because you have to basically hit every cell that is infected with the virus. In the case of hepatic delivery, or something, like previously I've worked on hepatitis C and hepatitis B, it may be possible to get a large percentage of the hepatocytes that are infected have delivery of an siRNA. That clearly is possible using AAV vectors, at least in rodent models. You can basically hit every single hepatocyte if you express the shRNA. But that's quite challenging.
So thinking perhaps about mRNA vaccines in general, what are some of the strategies or considerations that could be taken into account to generate a long-term vaccination effect, so to get memory T cell response?
I mean, I guess by the very nature of having an antigen expressed within the cell, as opposed to having the antigen come from the outside of the cell. Andy may be able to speak to this more than myself, but I believe that you get, you can get activation of both arms of the immune system, the humoral and the T cell, response from intracellular expressed antigen. But Andy may be able to elaborate on that a little bit more than me.
Yeah, I mean, there's evidence that we are getting memory responses already in the preclinical models. We have to remember that not all mRNA platforms were created equal. So there's different approaches. You've got, wild type RNA, conventional, modified conventional, self-amplifying. And so we're seeing, mixed data coming out, but there's definitely evidence that, we are already seeing a memory response. My expertise is around self-amplifying RNA. If you want to, induce better memory responses for those particular types of vaccine, allowing a longer interval between the prime and the boost is beneficial. For all mRNAs, you seem to need 2 vaccinations. And then the other part that we need to remember is the antigen. Not all antigens were created equal, and some of them are very immunogenic and tend to lay down, a memory in the immune compartment. COVID, or the spike protein, I think we're starting to learn that those responses wane somewhat, particularly with the base modified mRNA. We'll find out from CureVac and ICL in the coming months how theirs compare. This is certainly different than what we see with antigens like rabies, hemagglutinin from flu. We see a range of effects with different subtypes. H1 is very immunogenic and it seems to last a long time. The H7s, H10s tend to wane over time. So I think we have a lot to learn, but I think as we learn, the platform will innovate and we'll find solutions. So I think it's great that we're seeing with really the first push into the clinic with the mRNA platforms, we're seeing immune responses. We're seeing reactogenicity as well, both locally and systemic. But this is just the first wave, and we're going to learn through those human clinical trials, and the technology will adapt and move.
Okay, thank you. We have 2 live questions in queue here. We'll start with David, then go to Costas. So David, please go ahead.
Thank you. This question is probably for TriLink assuming 0.1 milligram messenger RNA per dose, what is the rough production cycle time? I mean, cycle time means from project process transfer to before release. How long does it take? Or what's the size of reactor volume for drug substance production? Assuming this is 1 million doses, either in one batch or in multiple batch based on the current technology limitation. So essentially that's about 0.1 kilogram drug substance we are talking about.
Sure. So to produce a 100-gram batch size, we at this time would probably recommend doing multiple batches, only because the amount of money that you are putting into that single reactor definitely causes some people to cringe. These reagents are expensive, as is the plasmid and all of the NTPs going into that. From the perspective of being able to produce 100 grams in a single batch, it's definitely feasible based on the reactor sizes available. But if you ask me to do this today, I would probably recommend breaking it down into, say, 10-liter batch sizes. From a 10-liter, you're probably going to get, upwards of 30 grams of product. So maybe 3 to 4 batches. And you can pool those for purification to create a single batch. It's just you're not putting all your transcription eggs in one basket. Transcription time is relatively short. 2 to 4 hours usually is sufficient for transcription, depending on the conditions that you've optimized for. And then the purification, we always are ideally, once transcription is finished, continuing all the way through. We don't want to keep any hold steps. The longer you hold RNA in either crude conditions or long-term freeze and thaws can definitely affect the purity. And so post-transcription, you would like to ideally go to a single purification and finish. Right now, we would estimate that time to be 3 to 4 days total. And so that's kind of what we're trending to as we envision our scale-up process, because right now we're not making 100 grams, no one's asked us to. And then your last question was around QC. It really depends on the QC release panel that you're looking for. The longest lead time items often are going to be bioburden testing. Those, you have to leave the product there to incubate for a few days before you can consider it to have a viable test result. But there are other QCs that also take a long time. For example, sequencing can definitely be a bottleneck. You'll be taking the RNA and doing RT-PCR followed by PCR followed by sequencing, and you'll have to optimize the primers. Once you have all of that stuff in hand, that could be maybe condensed to a couple weeks time frame. But the strategy really has been, in order to accelerate, once you've finished the drug substance manufacturing, you would proceed at risk to DP and then have the QCs from the drug substance kind of catch up to the DP release timeline so that you'll have your full package ready once you have your DP released. All right. Anyone else want to add anything?
Okay, if not, then maybe we'll move on to our next live question from Costa.
Hi, when you use multiple RNAs, does the encapsulation of each RNA is it homogeneous for each nanoparticle?
Yeah, that's a great question. So you would need to ensure that you are encapsulating to each RNA to the same extent. Typically, I think with the mRNA, LNP formulations we're performing here at PNI for clients or using our reagent Genvoy, we're getting very high encapsulation efficiencies, greater than 95%. So there's very little mRNA that's not encapsulated. If encapsulation efficiencies drop to 90%, you may then begin to see some differential or preferential encapsulation of different mRNAs over others within a mixture. However, that kind of raises another question, perhaps, is for the supply of your mRNA, you would have to provide a step where you would actually pre-mix your mRNAs prior to formulating into nanoparticles. So that's a decision that you'd need to make. In an R&D sense, you'd probably have them separate and then combine them just before formulating. From a GMP point of view, you'd have to think, do you have one GMP drug substance, or do you ask your mRNA vendor, GMP mRNA vendor, to make that mixture for you so you have a single drug substance?
So Lloyd, just a quick follow-on question then. So, you know, the siRNA LNP field has kind of led before the mRNA field. Is there any precedent there where they've mixed two siRNAs, encapsulated them, and you know what were the regulators asking there? It's not the same, but it's a precedent.
It complicates. I'm going to get into the stability and some of the QC aspects, right? It does complicate your testing. So if you have multiple RNAs in your particle, you'd have to basically not only show that they're encapsulated to the same extent, but also that over a stability that they are still functional, they are still intact. You can measure degradation profiles, and they may actually be different between RNAs. Even though they may have similar chemical modifications, they have different sequences. So that's something that you would need to show on with your stability data. And then you'd have the analytical challenge of actually resolving between two similar RNAs within your particle. So you'd need to resolve them. We can do that quite well, actually, with the lipid components in a RNA LNP particle. So typically, there are 3 or 4 or 5 lipid components in a lipid nanoparticle. And P&I has developed an analytical method based on UPLC that can actually resolve 4 individual lipid components and quantify them. However, you'd also need to do that with your RNAs if you have multiple RNAs in your particles. Another approach would be to have a mixture of lipid nanoparticles where each lipid nanoparticle, you'd formulate each RNA separately into a lipid nanoparticle, and then to basically present multiple RNAs, you'd actually take 2 different LNP formulations and mix those. The only downside of that is that you'd have to make an assumption that if you need, say, there's 2 components, a long RNA and a small RNA, and you need both of them in the cytosol at the same time, you'd have to ensure that you'd have those 2 separate particles at the same location at the same time within the cell. So by co-encapsulating both a long and a short RNA in a single nanoparticle, you are basically increasing your chances of delivering those multiple RNAs to the same location at the same time.
Thanks, Lloyd. So Shell, what's the next question?
Yeah, so sort of a follow-on question about co-encapsulation, maybe more for Annie here. If you were to design, say, slightly different mRNAs encoding slightly different antigens, would there be any benefit to doing that and then co-encapsulating those in a particle, for example?
Yeah, certainly. You know, for instance, you know, if you wanted to generate an immune response against 2 different parts of a virus, you might have an intracellular protein, a matrix or a nuclear protein that you wanted to generate a T-cell response against, and then you have a glycoprotein antigen. You could mix those 2 together and use that as a co-vaccination strategy. Obviously, we've been through if we mix them together, that adds complexity, but you could have them separately formulated and then add them together. One of the benefits of the self-amplifying system is you can do that already. That viral genome, after the subgenomic promoter, is designed to express, for the alpha virus, the E1, E2 glycoproteins and the capsid. And so there's plenty of preclinical data showing multiple antigens being expressed off there. But I think Moderna has shown, 5 antigens from the CMV pentamer can be, put into one vaccine and expressed as that membrane complex, and immune response is generated. So there's certainly precedent for doing that.
Annie, did you have any thoughts on that coming from your less from the RNA perspective, but more from the traditional antigen design perspective?
So we've seen that it's popular for our two-component protein nanoparticles, or it's possible, where we bisostronically express two different genes for our nanoparticles, and they do assemble within the cell. The other avenue that I could see this going is we are developing protein-based adjuvants. So potentially in the future, we could deliver both an adjuvant and an antigen together as an mRNA.
And that's actually a good lead-in then to another question we've had a few about is there any work in adding adjuvants to mRNA vaccines? You know, what are we seeing out there in terms of people needing to or choosing to add adjuvants to their vaccines? And I should add, this is a question coming from one of the breakout sessions earlier.
Oh, maybe I'll take that. I mean, yes, there's already people starting to try and express, you know, adjuvants off the RNA, as Annie highlighted, but also, you know, people are starting to try and add different lipid components to the LNP to aid in adjuvant responses as well. Obviously, there seems to be, you know, both local and systemic reactogenicity in a dose-dependent manner to the RNA vaccines. And so as you go up the dose, you know, the clinical data we're seeing for the current coronavirus, you know, around that 100 microgram and above range for the base modified RNA, we're seeing adverse events. And one of the ways we might see of bringing that dose down is to find a different way of adjuvanting the formulation. Certainly the doses that are being contemplated for CureVac and Imperial College with wild type mRNA seem to be much lower. I haven't seen anything published about the dose from CureVac, but for Imperial College, their published clinical trial design, they're dosing at, you know, 0.1, 0.3, and 1 microgram. And so I think we're going to see the next generations of vaccines looking to try and lower those doses. And one of them may be adding both a, you know, a small molecule type adjuvant or including it in the mRNA.
Great. Then perhaps we can move on to the next question. There's been a question in the chat that someone has been trying to ask. The question is about what type of 5' cap is most suitable for an mRNA vaccine?
So I think that may depend on whether or not you are doing an mRNA vaccine versus a self-amplifying RNA. So we know that in, at least in mouse liver, that the duration of expression of a cap0 RNA is very short. So, and you get longer duration of expression from a cap1 RNA. So I would say that if you're going to do an mRNA vaccine, you absolutely would want it to be a cap1. In terms of the alpha viruses are actually cap0 in their native form. We did do an experiment with Kurt Kamrud at Synthetic Genomics, where we compared cap0 and cap1 RNAs in the context of a self-amplifying RNA. And there did seem to be some noticeable improvement using a cap1 RNA, but due to the small number of mice that was used in this experiment, those results were not statistically significant. And so I believe the jury's still out on that one with respect to a self-amplifying RNA. I don't know if Andy has any experience with this from when he was at Novartis.
Yeah, I mean, certainly what we found from Novartis was, you know, when we're using an LNP delivery system for the self-amplifying RNA, when the readout was immunogenicity, there was no difference between cap0 and cap1. There were slight differences in protein expression, the cap1 being slightly better, but it had no impact on the immunogenicity. And so I think both work quite well. No data in humans though on the relevance. It was all, you know, preclinical mouse.
Yeah, I mean, what I showed you, and for those of you that joined us for our breakout session, is that we can use this co-transcriptional capping method called clean cap to put a cap1 structure on the end of a messenger RNA in a very highly efficient manner. You can actually make a clean cap that is cap 0 to intentionally have a cap 0 RNA. I would say, given the fact that we're now on a path to producing kilograms of clean cap 1, that if there were no difference just from a cost of goods perspective, you might want to go with the cap 1 RNA simply because that's being mass produced for other people's applications.
So one question we've had is regarding the locations of manufacturing, and specifically, are we seeing any, is it possible to set up everything from your mRNA synthesis through to your fill finish in one site? Or are these occurring at different locations, and are you having to arrange transport between them?
I can speak to TriLink's capabilities. Currently, we are only doing drug substance manufacturing, no drug product here. So we do arrange for logistics to transport to another third party that does the formulations. I think, obviously the strategy would be for a commercial application, if we could condense that to a single site, would be very advantageous.
Yeah, I mean, I'll make a comment here. I mean, I think, the technology's in its infancy. The current pandemic, we've seen an acceleration of RNA into the clinic because of the production capacity. So, right now, drug substance, the API tends to be made, at a dedicated facility, and then the formulation process happens, somewhere else. It's only natural that we're going to start to see that being combined and co-located. I think that's, certainly happening. That's already done at CureVac, Moderna, BioNTech. But, for the rest of the field, it's still, fragmented to some degree. But given the volumes and size requirements, it's still doable, and we'll see the field evolve over the coming years. I don't know if you have a comment, Anton.
Yeah, so I mean, right now, we do manufacturing at TriLink for certain phases, clinical phases, but it's actually relatively common for us to arrange for tech transfer in the same way that P&I is, shipping their instruments to third parties to do manufacturing. And so, from a customer perspective, there's not really anything that would prevent this from all happening in one location. So we do both mRNA manufacturing, but then we also provide clean cap and other reagents such as modified NTPs to third parties who do their own manufacturing and could potentially have one of P&I's instruments sitting right next to, one of, a bioreactor making messenger RNA. All of these technologies are relatively low footprint. Really, they could happen in a single or a couple different rooms. And so it's pretty straightforward to put this all into one flow. And there's been a movement in the field to kind of integrate different parts. It's something that we didn't really touch upon very much in so far as there's plasmid sourcing. And so I think eventually this is going to be all integrated into a one-stop shop. But everyone's running as fast as they possibly can right now. And so rather than working on the vertical integration, we're working on expanding our scales and capacities horizontally. And so it would be desirable to do that. But at this time, we're mainly focusing on scale up of the mRNA process rather than vertical integration.
And certainly, in the UK, we're seeing, the UK government trying to establish infrastructure both for making RNA and the formulation process. In Canada, P&I is in discussions, with the Canadian government to build a capability to produce mRNA in Canada, for, this pandemic or potential future pandemics. We're starting to see, countries around the world really consider, do they have the capabilities in country to do this? And they're starting to realize that, RNA vaccine technologies provide that opportunity to do that at quite a low cost and a small manufacturing footprint.
Thanks. And sort of following on that, there have been several questions about the analytical aspects of this. And so I wonder if there's, what are the in-process analytics you'd have to do for manufacturing, say, an RNA? And then maybe, Lloyd, you can comment also on what you might need for an LNP. And is there any overlap between these? And is there some economies of scale that could be had by vertically integrating these?
Yeah, well, the support, the quality systems, QC, QA, analytical methods, a lot of the analytical methods you need for, say, the RNA drug substance, you need those methods actually for analyzing the RNA within your nanoparticle drug product and finished product. So there's certainly some savings there. Since both technologies employ relatively small footprints, a single facility is probably preferred. And again, if you are thinking about multiplying those, copying those facilities, having a blueprint that you could basically build similar facilities around the world, you basically have a complete solution there, a one-stop shop.
And Jessica, I think you had something you're going to add to that.
Oh, I was just going to comment on in-process assays for RNA. I mean, from our perspective, we're monitoring the process to make sure that quality and yield are sufficient. So really, we'll look at increasing those panels of in-process QCs as we increase, or as we go to later phases and do some more robust process validation. But right now, it's typically just purity and yield monitoring throughout the process, and then some bioburden, obviously, as well.
And that's done primarily by HPLC or?
It really depends on the method that we're going to be using for the final release. And so if we're using, say, a fragment analyzer as the method of purity for the final product, we would use fragment analyzer in process. If it was an HPLC, we would do HPLC in process.
Thank you.
So we focused a lot on lipid nanoparticles today, but as Yvonne mentioned, there's a number of different delivery technologies. So how would some of these considerations we've been talking about and process steps we've been talking about, how would they compare for example, a polymer-based system?
Quite similar. The actual drug product or LNP, the nanoparticle manufacturing steps may obviously be different. Your input materials to make those polymeric nanoparticles will be different, but the same idea of in-process testing, key process parameters, the need to sterile filter and vial your final product, those things are the same. And obviously your input RNAs as well. So there are a few differences, but the overall manufacturing process flow does not change appreciably, maybe with the addition of 1 or 2 steps.
Great, thanks, Lloyd. I guess maybe everyone can chime in a bit about whether or not, or what might change about their part of this, their piece of this puzzle, if we change the route of administration. So if you're designing something for intramuscular versus subcutaneous, how does that change your antigen design? How does that change your mRNA modifications? How does that change your lipids?
I mean, I can speak to the mRNA modification part. I think that it would be largely similar by those 2 different routes. I mean, I think that depending on what your dose and your route is, maybe the more important thing is what you choose as your purification. We're seeing, for example, with the Moderna, that there was some toxicities associated with higher doses and things like that. And so I think that monitoring double-stranded RNA, for example, is important. And so depending on what your application is. In terms of the previous question about formulation, I mean, one route that we haven't really talked about is that a lot of people are using messenger RNA for doing engineering of cells, in which case you might use electroporation as your route of administration for an ex vivo treatment. And so in that case, you know, you would want it to be in an electroporation media when you're, you know, treating the cells. So that's another consideration. We didn't really touch much on that today since this focused more on vaccines, but a really large area of which messenger RNAs are being applied is, for example, engineering of CAR T cells and things like that for immunobiology.
Yeah, if I'll make a comment, you know, on the LNP, you know, certainly the work we did at Novartis and published in the mouse model, we tried, you know, intradermal, intramuscular, subcutaneous vaccination, and we found that intramuscular worked best, and it utilizes standard syringe and needle that's readily available. The reactogenicity, local reactogenicity tends to be better as you progress, you know, towards the clinic. But there is precedent. But with the LNPs we developed, we saw no evidence of antigen expression in antigen-presenting cells, dendritic cells, but certainly in the literature, you can do that, but the formulation tends to be different. And it's a different way of presenting the antigen to the immune compartment. And, you know, intradermal vaccination seems to work well, but it looks like the formulation is different than those used for intramuscular vaccination. I think it's early days, and those self-amplifying systems don't tend to work that well, that route, but mRNA, there's good evidence that you could express an antigen in the immune compartment and generate an immune response that way.
Shifting gears a little bit to back to, I guess, a bit of some of the manufacturing challenges. What are some of the supply chain risks that we're seeing, especially in this pandemic situation right now, from start to finish for manufacturing an mRNA LNP?
Well, the two main components of the LNP would be the RNA and your lipids. So I can speak a little bit to the lipids. The majority of the lipid components are already available GMP from multiple suppliers. I don't know their current inventory levels, but I know the demand has increased for those commercially available GMP lipids. The second part of the lipid equation is the custom lipid or custom ionizable lipid that so many people are using in their formulations. Not everyone who's working with lipids are using ionizable cation lipids, but the vast majority of studies do. You are in two situations. One is you already have a manufacturer of your ionizable lipid under GMP, or two, you're trying to find one. So I think the people who already have identified vendors to manufacture their custom lipid components are in much better position than those who are trying to find capacity. Because I've talked to a number of lipid vendors and they are full up. And I can't speak for the RNA manufacturers, but I'm sure you're in a similar position as far as your kind of part of the industry goes.
One thing that I think was very beneficial for TriLink is that we moved into this new facility in January 2020 that allowed for 5 parallel manufacturing tracks. Last year we had 1. And so if we were in this case last year, it would be a much more longer turnaround time. So right now, you know, there definitely is a queue, but not super extended. Oftentimes we can align our queue availability to the availability of the reagents. So kind of looking back on where our bottlenecks tend to be is definitely around the plasmid manufacturing. That seems to be a huge bottleneck for every program for us, is getting that plasmid in hand. I think Bejoyta and Brett spoke a lot about their scalability, and we haven't experienced any enzyme shortages, so that's a good thing. but definitely plasmids is the biggest thing. And then there have been a few instances of like difiltration cassette cartridges that are used across, you know, the drug substance, the drug product, and all the different manufacturers, and not just for RNA. I know those have been in high demand. Nothing considerable that has put us in, you know, a stop manufacturing situation, but, you know, securing supplies and keeping our vendors on top of what we need has been critical. And then the last thing is just resins for purification. There are a few resins that a lot of people are using for mRNA manufacturing, like poly-DT resins. I know that those are in high demand right now. So again, it's just working really closely with the suppliers of those to make sure that we have what we need when we need it.
So I just to follow on to that, I had a quick question for NEB. Obviously, there's a lot of research activities on mRNA vaccines. Have you got supply problems with the kits and the reagents for that kind of research base, or are you able to stay on top of it?
So maybe I'll take a stab at it, but Joyta can jump in. We've not had issues in supplying, you know, sort of our research customers. A lot of them around the world were sort of shut down and are just starting to come back now. The issues were, you know, as you'd expect, there were shortages of sort of small scale, research scale RNA purification columns. Because everyone was trying to purify RNA to put into their diagnostics tests. But those have, I think, largely been solved, and I don't think anything had gone on back order to support either the research or our larger sort of OEM customers over this you know, 6 or 7 months.
Okay, so I think we have time for maybe one last question, and I think it is poignant, you know, at this point in time to ask, you know, how far are we from getting to a vaccine for COVID? Maybe everyone can chime in a little on where they feel that is.
Well, so I'll kick off. I mean, it's certainly, you know, conventional technologies. We've got a couple of viral vectors already with data. They're generating immune responses. Whether it's going to prevent infection or prevent severe disease, we'll see. The first mRNA vaccines are pretty reactogenic, both locally and systemically in a dose-dependent manner, but we're seeing immune responses. It'll be interesting to see how, you know, we've got 2 other forms to come in terms of data, the CureVac approach, conventional without base modifications, self-amplifying. You know, from Imperial College. So I think for the mRNA side, it's encouraging, but, you know, the full platform spectrum still needs to come out. Immunogenicity is the early readout with safety, and then we've just got to see how RNA performs in the efficacy side, and we'll be able to see how it compares to traditional subunit viral vectored vaccines.
I would say that I'm relatively optimistic that we're going to get some level of immunity from some of these vaccines. Whether or not it'll be durable or not is a really open question. But I think that one of the really appealing things about the mRNA platform is that in theory, you should be able to re-administer if this becomes a seasonal thing, that, you know, you should be able to give this as, you know, as frequently as you really need to. And the doses are incredibly small. And so I'm relatively optimistic, but I'm not a vaccinologist. So.
Okay, Andy, I think we're ready to close this one out because I think Frank is on board and
Perfect.
ready to start talking.
Okay, so we're going to close out the session just with a few words from Frank Zlak, who's director of the Institute for RNA Medicines at Beth Israel Deaconess Medical Center there in Boston. So, you know, he's going to give us a few words on the future. Over to you, Frank.
Thank you very much. I'm just looking to see how I can share my screen here. Desktop, share. Just give me a second whilst I do this now. Oh, right. Hold on, I'm having an issue. Let's try again. We did practice this, and for some reason,
it's not
right
working.
now, I'm having a problem. Actually, Jane, do you have my slides that you can just show? I think I might have sent them to you.
Yeah, Jane's going to try and pull them up for you now, Frank.
Yeah, I don't know why. It's suddenly giving me some thing that says advanced versus basic, and I have never seen this before. I just upgraded to the new Zoom a few days ago, so maybe this is a new feature, but so I'm sorry. Anyway, maybe I can just start by saying thanks for the opportunity to be with you today. I was asked by Precision Nanosystems to close out this symposium by trying to give you a sense of the broader impacts or the broader features of RNA medicine. We've heard a lot today. We've heard a lot over the last few months about mRNA, RNA medicine. We've heard a lot about vaccines, mRNA-based vaccines. We've heard a lot about delivery of mRNA. But one thing that I just wanted to try to impress upon you today in the next sort of 15 minutes or so is that the RNA medicine space encompasses many different classes of RNAs. So what I will hopefully be able to tell you a little bit about today is some of our work in trying to harness some of the discoveries in non-coding RNAs for new personalized medicines. And the example that I'm going to give you today is microRNAs, but much of what I tell you about today is going to be true for other classes of RNAs as well.
Frank, I have your PowerPoint up too, just so you'll know. So you can just tell me and I'll start working it for you when you're ready.
Thanks, I see it. That's great. Yeah, so just by way of disclosure, I'm on the scientific advisory board of those companies shown there in the middle bottom, and I'm also on the advisory board for Precision Nanosystems. So if you can go to the next slide, please. So before I begin, let me just say thanks to all of you guys working on vaccines, and thanks to all of our frontline healthcare workers for putting their lives at risk and so helping so many people through this pandemic. Next slide, please. So there's, of course, a lot of excitement around non-coding RNAs. Non-coding RNAs make up sort of a greater proportion of RNAs in our cells than do the coding RNAs. And of course, we've heard a lot about protein coding RNAs today and trying to harness them for new vaccines or for protein replacement. But there are probably 10 times as many non-coding RNAs in our cells. So if you go to the next slide, these have provided a paradigm for gene regulation and of course a new paradigm for therapeutics and diagnostics. The other thing I wanted to say is that RNA medicine is undergoing a renaissance. We've heard today about the efforts in the vaccine space, exemplified by Moderna here just across the street from us, and that's in the bottom right. But of course, RNA medicine encompasses trying to target protein coding RNAs, trying to target non-coding RNAs, for example, using siRNAs or antisense technologies. And those are exemplified by the successes of Alnylam in the RNAi space and Biogen and Ionis in the antisense space, shown there. In the next slide, I'm just going to point out that RNA medicine is becoming highly personal. And many of you are probably thinking about personalized vaccines or personalized cancer vaccines. But just to give you another example, this is an example that came out just a few months ago from Boston Children's Hospital, from Tim Yu's lab, where they designed an antisense molecule that would allow splicing of a mutation, or that would allow splicing around a mutation in a patient that has Batten's disease. Now, as far as they knew this patient was the only one in the world who had this particular mutation, and they were able to design this oligonucleotide that was personalized just for her mutation. And so this kind of strategy now of being able to design personalized RNA medicines is just beginning, and I think it's going to become way more important in the future. So next slide, please. So just to illustrate why I think the non-coding portions of our genome are so exciting for new therapies, of course, the genome sequencing project, human genome sequencing project, and the ENCODE project showed us that we had many genes in our genome, about 20,000 protein coding genes. But the ENCODE project showed us that we have many transcribed regions of our genome. And in fact, if you just pushed the button once, they showed us that there were many different classes of RNAs that were being expressed from our genome. We know of at least 2,500 of these small RNAs called microRNAs. There are many more isomers, which are sort of variants of those microRNAs, maybe up to 250,000 variants of microRNAs made within our cells. About 100,000 piRNAs made in our germline, which protect our genome from transposons and the like. If you just look at this pie chart here, it shows you that our coding genes are now being dwarfed a little bit by our non-coding genes. There are at least 48,000 long non-coding RNAs, almost twice as many as there are protein coding genes. All those pseudogenes, those 20,000 pseudogenes, the vast majority of those are expressed, and there are examples now of them having functions in cells. 100,000 or more circular RNAs. So these are examples of how of sort of back-spliced RNAs, which instead of the forward splicing that we've heard a little bit about today already, there are examples of back splicing that make a circular version of the RNA. And then all of those tRNAs that are functioning not only as as an important part of translation, but also that get fragmented into smaller fragments and have functions. So if you add that all up, that's why I come to the number of about 10 times more non-coding RNAs than there are protein coding RNAs. And of course, this is a lot of complexity, but with this complexity comes a lot of opportunity. We now have many, many more ways, I would say 10 times more ways, to try and affect disease or to understand disease by trying to understand what are the contributions of the non-coding RNAs and trying to target the causal non-coding RNAs as therapies in those diseases. So next slide, please. So just to reiterate that this provides us with some opportunities for novel therapeutics where the RNA can be the drug. We've already heard a little bit about that today, where you can try to restore a protein or you can try to introduce a vaccine. I've used the example here of restoring a tumor suppressor RNA. In the second example, the RNA can be drugged. This could be using an siRNA or an antisense approach. And in this case, the example I've given is that you might want to inhibit an oncogenic RNA. And then lastly, the biogenesis of these RNAs can also be drugged. So in the next slide, I've just tried to give you a sense that the number of clinical trials or the sort of the involvement of non-coding RNAs in medicine is on the rise. I've just shown you here, for the interest of time, a keyword that I did on clinicaltrials.gov for microRNAs, and you'll see that there are 890 clinical trials involving microRNAs. That number goes way beyond 1,000 if you look for all the classes of RNA medicine that are out there. So we're definitely in an upward trajectory. We expect that many of these molecules are going to make their way into the clinic at some point and will be helping patients in numerous different diseases. So the one example that I just wanted to highlight today, if you can go to the next slide, is a clinical trial that began following some of our preclinical work, showing that particular microRNAs can be oncogenic. So this is an example of a microRNA called miR-155 that we showed many years ago now was altered in expression in many B-cell lymphomas and leukemias. We overexpress miR-155 in mice, and you can see in the top left there that the mice get lymphomas. That mouse on the very top left has a huge lymphoma growing out of its neck, an axial lymphadenopathy. But when we target miR-155 with an antisense molecule, we could essentially cure that mouse from lymphoma. So work like this, if you go to the next slide, led to a collaboration with Miragen Therapeutics. And Miragen Therapeutics has an anti-MIR-155 molecule called cobamycin, also known as MRG-106, that's in clinical trials for patients with different kinds of lymphomas. And from what I understand, the safety profile of cobamycin is very good, and this molecule is now in phase 2 trials for efficacy. So next slide, please. So I'm just going to end by telling you that at the Harvard Initiative for RNA Medicine, we obviously want to make and translate RNA discoveries into novel therapeutics and diagnostics. So in the next slide, we have a number of different agendas or initiatives. We really want to try and understand the component of RNA in disease, the different components. We need to discover which RNAs are involved in disease. We need to understand which ones are actually causal in the disease and which ones might be novel biomarkers. We ultimately want to integrate these RNA, this RNA information into the patient standard of care. So potentially offering novel RNA-based therapeutics that are unique to each patient. And then of course, we want to deliver RNA-based therapeutics in an effective manner. And what I'm going to talk about on the next slide is that we've started a core facility here at Harvard with the help of Precision Nanosystems. We've installed a number of, well, all of the NanoAssembler products. And anybody who's interested in trying out some of these NanoAssembler equipment for delivery of different RNAs, welcome to contact us. We are open to non-Harvard and to non-academic institutions. So in the next slide, I'm just going to end with one example from our lab about how we've taken this idea of trying to understand non-coding RNAs and harness them for potentially personalized RNA therapeutics. So this begins with a collaboration between my lab and Dr. Mani Hidalgo and Senthil Muthuswamy here at Beth Israel Deaconess Medical Center. They had not only RNA samples from a variety of different pancreatic cancer patients, but they'd also grown pancreatic cancer organoids or avatars from each of these patients. So there were about 30 different patient avatars that existed here at BIDMC. So we were able to identify all the RNAs that were misexpressed in those patient avatars. And then because we had the avatars, we could ask whether any of those RNAs were causal or potentially causal in the tumor phenotype in those avatars. So if you go to the next slide, please. This just shows you on the left that of those 30 tumor samples, some of them had high levels of MIR21 expression. So I'm just talking about MIR21 in this example, but of course we profiled all of the microRNAs from these samples. But MIR21 was illustrative of the point that I want to make today. But you'll also see that some of the patients had normal levels of MIR21 in their tumor cells. Now, MIR21 expression has been shown to be prognostic in pancreatic cancer. So patients with high MIR21 expression generally have a poorer prognosis. So it looks like these patients are sort of sorting into a category with high MIR21 and those with lower MIR21. And this allowed us to ask, was it possible that an anti-MIR21 therapy might work in patients with high MIR21, but maybe would have no effect in patients with low MIR21 expression? So next slide, please. We already knew that MIR21 was an oncogene. So in work that we published a few years ago, we overexpressed MIR21 in mice, and we found when we express it in the hematopoietic system, we get a B-cell lymphoma, pre-B-cell lymphoma emerging in these mice. That was also dependent on the expression of MIR21. So MIR21 looked like it was an oncogene and that the tumors were addicted to this particular oncogene. Next slide, please. So we used a sort of a nano conjugate type system, sort of a nanolipid conjugate type system shown on the top left to try and target MIR21 with an antisense molecule. So this was work we did in collaboration with Sangeeta Bhatia's lab at MIT. And what I'm showing you on the top right is that if you label the antisense MIR21, we can show that the MIR21 actually gets into these pancreatic cancer organoids. So organoids are those spheres that you see in the DRC image at the sort of in the middle of the slide. On the bottom left, I'm showing you that those organoids are dependent on expression of miR21, because if we deliver miR21 into those organoids, on the very bottom left, you can see the organoids start to disintegrate, and we see increased apoptosis, which is the red dye that you're seeing there. I'm not going to show you today, but we injected this into the tail vein of mice, and we could deliver it to tumors. Of course, we get a lot of delivery to liver, which is an issue that many people see with nanoparticle delivery, and we're working on better delivery vehicles for this kind of strategy now. But we were able to see delivery to the tumor. And of course, those tumors grew more slowly, which I'm not going to be showing you today. In the next slide, I just wanted to talk to you a little bit about this concept of personalized anti-miR21. I told you that there were some cancer patients that had high levels of miR21, including PDO286 and PDO219 that had excess levels of miR21, and a few that had normal levels of miR21, and those are exemplified by PDO017 and 030. Shown on the top right is a graph of the quantification of miR21 expression. And what we were able to show is that only the tumors that had high levels of miR21 would respond to an anti-miR21 therapy, shown in the middle where it says high miR21. So the third panel in that middle section are the organoids treated with the anti-miR21. And the organoids grew to a smaller size, or they started to undergo apoptosis. In contrast, the patient that had normal levels of miR21 did not respond to the anti-miR21 therapy. Funny enough, this patient did respond to gemcitabine. So this is a second approach that BIDMC is now taking, is to treat these organoids not only with microRNA-based or RNA-based therapies, but also with drug-based therapies to sort of pre-screen the patient's organoids to see which drugs might actually work for that particular patient. So in the next slide, I'm just going to conclude by saying that in this part of the work, I hope I've told you that RNA provides an opportunity for personalized medicine. In many cases, the RNA is the companion diagnostic and the therapeutic target. So if you can identify an RNA that you know is causal in the disease, then its expression is the diagnostic, and going after that RNA is the therapeutic. You might be wondering about some of the other organoids that had low levels of miR21. Well, they have altered levels of other microRNAs, and so we're trying to design strategies where we can now test whether those patients would be responsive to a different microRNA. So that's the concept of this personalized idea. Of course, this is kind of a trivial example that I've given you, but we hope to expand this into a more concrete sort of platform where every patient might one day see an avatar and a potential RNA-based therapeutic. Next slide, please. So I'm just going to end by thanking the members of my lab who performed some of this work. I'd like to thank my collaborators and you for your attention. And please let me know if any of you want to try out some of the precision equipment. You're welcome to come and test it out in our core. And with that, I'm going to close. Thanks very much.
Okay, thank you very much, Frank, for closing us out. We're right on time. I'd just say to everybody, we're going to endeavor to continue to answer your questions. We've still got many to answer. We'll get you answers, online. We will, the recordings will be available for view after the seminar. So with that, I'd like to say thank you for everyone who attended. Thank you to all the presenters. Hopefully, the audience has learned a lot, and we've encouraged you to either continue in the RNA medicine space or venture in with your work into a new field. So with that, I'd say thank you and close you out.
Thank you, everybody. Bye.
We'll stay on just for a few minutes just to make sure if you have any questions, you can still put them into the Q&A. So we'll just stay live for just a few minutes to allow you to add any other questions you want to ask. Remember, if your name is associated to your question, even if we didn't get to it, we'll follow up with you and get answers to that question. So all of our speakers have agreed to, helping us with getting answers or getting you connected to the right resources. Also on our webpage, we are going to have where we had like the programs and stuff, we're going to have a page for resources. so that all the different speakers that you heard from TriLink and NEB, from Frank, everyone else kind of has links to the things that they reference. So you could be able to click onto that and get onto their website or to get onto different areas that they'd like to, that they reference during their presentations. And then finally, you can expect in the next probably about a week to 2 weeks. So give us a little time because it's a lot of video. We're going to put together the recordings and we'll send them out to you. Not all sessions were recorded. Some of our breakout speakers did not, were not in a place where they could allow their sessions to be recorded. Other than that, thank you all for joining us. And again, ask any questions you want. We have some things coming up in September, so you'll look forward to seeing more that we're going to do some webinars on regulatory and manufacturing with the CMO panel. And we appreciate your time today. Thank you.